செக் பண்ண விட்டு பார்த்தோம். விட்டு பார்த்தோம் சார். மோட்டர் ஆஃப் பண்ணி தான் பார்த்துட்டு இருக்கோம். ஆஃப்ல வந்து சார் டெமோ மோடு. ஆமாம். அதான் அந்த டெமோ மோடு என்ன ஆகிடுச்சுன்னா அது ஆக்சுவலாக வந்து கேமரா சேவ் வச்சுருப்பாங்க சரிங்களா. அப்போது டிஸ்பிளேல கனெக்ட் பண்ணி வச்சுருப்பாங்க. ஆட்டோமேட்டிக்காக கேமராவோட ஃபீச்சர்ஸ் எல்லாம் வந்துச்சு. அதனால தான் கேமரா உட்காந்து ஸ்லீப் மோடு போகும்போது ஆட்டோமேட்டிக்காக ஆஃப் பண்ணி வச்சு
இவ்வளவு நேரம் கூட இல்லை சார். இது இவ்வளவு நேரம் கூட இல்லை சார். இல்லை இவ்வளவு நேரம் கூட இல்லை.
ஆமா, எனக்கு மட்டும் 5.5 மணி நேரம் ஸ்டூடியோ இருக்கு சார். 499 பேர் பார்த்துருக்கேன். ஆனால் இந்த ஹோமோட கேமரா ஐடியில் ஆகும்போது கேமராவோட ஃபீச்சர்ஸ் டிஸ்பிளே பண்ண ஆரம்பிச்சு.
ஸ்டோரேஜ் கம்மி தான் சார்.
என்ன சார், ஆண்ட்ராய்ட் ஃபோன் கொடுக்குற அப்டேட்லேயே ஃபோன் ஃபுல்லாகிட்டு அப்படியே 8 ஜிபின்னு சொல்லிடுறாங்க. கடைசியில் பார்த்தா ஒரு 1.5 ஜிபி நம்ம தான் ஸ்பேஸே காலி ஆகிடும்.
I request everyone to keep your mobile phones in silent mode. Thank you. Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world, said by Louis Pasteur. A very good morning to everyone gathered here. On behalf of the organizing committee, I extend a warm welcome to all participants for Day 2 of the National Seminar on Biotechnology-Driven Solutions for Aquaculture Farming, Nutrition and Feed Efficiency. We hope today's sessions will be equally insightful and academically enriching. With this note, it's my honor to welcome Dr. R. Subhashini, Assistant Professor, School of Biotechnology, to introduce the resource person of Technical Session 4, Dr. P. Sandanam, Professor, Department of Marine Science, Bharat University, Tiruchirappalli.
Good morning, everyone. It is an immense pleasure to welcome our esteemed chief guest, Dr. P. Sandanam, Professor, Department of Marine Science, School of Marine Sciences, Bharat University, Tiruchirappalli. He completed his MSc, MPhil, PhD in Marine Biology from the Center of Advanced Study in Marine Biology, Annamalai University. His field of specialization is marine planktonology and aquaculture. His areas of research expertise include biodiversity, nanobiotechnology, bioremediation, and bioprospecting of marine plankton, mass production and application of marine plankton as live feed in aquaculture, and sustainable aquaculture practices such as biofloc, aquamimicry, and aquaponics. He has over 20 years of teaching and 20 years of research experience. He has received several awards, honors for his contribution in the field of science, such as Young Scientist Award by DST Government of India, Best Researcher Award by KTSTM Trust. He is also a scientific advisory member for Rajiv Gandhi Center for Aquaculture, MS Swaminathan Research Foundation, Reliance Foundation, Marine Product Export Development Authority, and in various other reputed sectors also. Dr. P. Sandanam has received totally 50 major research projects funded by various funding agencies like UGC, DST, DBT, MOF, and CC, MOS, SERV, ISRO, Government of India, FANCY, Government of Tamil Nadu, and ECOSOC, United Nations, with a total budget of 6.06 crores. He successfully completed 10 major research projects and currently running 5 projects. So far, he has published nearly 286 research articles and submitted more than 80 gene sequences on microbiology. He has so far guided 9 postdoctoral fellows, 20 PhD candidates, 14 MPhil, 1 MSc, and 3 BTech students, and currently guiding 1 postdoctoral fellow, 8 PhD candidates, and 3 MSc students. He has visited countries like Dubai, State of Eritrea, and Yemen. He has delivered so far 250 invited lectures in various conferences, trainings, professor courses, etc. He has developed the technology for the large-scale production of marine copyfoods for the first time in India. He established live feed culture, RAS, bioflag, aquamimicry, aquaponics facilities in the vicinity of Bharat University. The book entitled Advances in Marine and Brackish Water Aquaculture, edited by Sandanam Mehta, published by Springer, is rated as one of the best marine biology books of all time in the world. He is a distinguished professor in the field of marine science who has made significant contributions. We are truly privileged to have such an inspiring personality among us today for a national seminar on biotechnology-driven solution for aquaculture farming, nutrition, and feed efficiency. Thank you, sir.
Thank you, ma'am. I now kindly request a speaker to commence the technical session on the topic Sustainable Aquaculture Technologies, Fostering Economy, Environment, and Employment.
good morning. At the outset, I would like to thank the authorities of this esteemed college, especially the correspondent, the visionary man, Dr. Batmanathan sir, the secretary of this college, Dr. Geeta ma'am, principal, Dr. Sumati ma'am, associate professor and head, Department of Biotechnology, other faculty members of the department, my own colleague, Professor Rajaram, and my professor and head, Professor Damodaran, the faculties from other college institutes, research scholars, students, and my dear participants. Very good morning, Manada. So today, I hope you all enjoyed yesterday's session, which covered the fish culture farming and feed formulation and nutrition, and an overview of arena of marine science opportunities in marine science and fisheries. So today, let us start with this lecture, my lecture on sustainable aquaculture technologies, fostering the economy, environment, and employment. Now, recently, the Department of Biotechnology, Ministry of Science and Technology, Government of India, they launched this special program. You just visit the website, you can get a BioED. There is a special program called BioED. They are regularly giving call for proposals for your students, faculties. If you submit any proposal on economy, environment, and employment related proposal using the biological organisms as a raw material, so you can be sanctioned project. So they have plenty of money in the ministry, so that they launched the BioED. And the Sustainable Development Goals, United Nations, they have a lot of 17 SDGs, under which the food scarcity, poverty, malnutrition, this all comes under the SDGs. And as a marine science or marine biology, or of course aquaculture or zoology, of course all biological science which is relevant to marine or aquaculture or aquatic ecosystem, which is classified under the 14th goal. That is life below water. How the life below water supporting for economy of country, how they provide the employment, and how they conserve the environment. This is the concept behind the SDGs and also the BioED program. So you please visit the biotechnology, the website Department of Biotechnology, and we have a Ministry of Earth Sciences. They launched the Deep Ocean Vision, a specialized program scheme called Deep Ocean Vision, starting from microbes to mammals. If you have any project, if you have any concept with you, you can submit any time. They have plenty of money, and they can provide the fellowship also for the students, master's degree students. They provide the fellowship, and they provide the postdoctoral fellowship also, those who are completed PhD. And for projects for the faculty. Why I am telling this for, we have a lot of scopes, but we have to go through the websites which is relevant to your subject or science, so that you will get a lot of idea or information. You can shape your future. Okay? So we have a very good opportunity. Even yesterday, we have discussed with some students from the same department, biotechnology. I really very surprised to see that they published 3 papers, one in elsewhere and another one in spring care, that is on cellulose production from the seagrass. A student from this college, college from Biotechnology department, they published 3 papers that took from seagrass, using seagrass they developed cellulose, they extracted cellulose, and they developed some package material, which is biodegradable nature. It is environment friendly. So this is the need of the hour. Now the funding agency, they are looking the proposal which has societal relevance or national importance. You just like you should not submit the proposal related for publication and board. They never turn to your side. They are looking for product patent, and that your outcome of your project should ultimately reach the society immediately. So that they started a lot of startup programs, incubation centers. You should see that website. Regularly they are being, now it's also called for the Department of Biotechnology website. That's BioD3. Lot of schemes are available, fellowships are available. You can make use of the available funding facility for your future career, so that you will become a dynamic academician or a scientist. You are like. So with this, let me start the today's business. This is the title I have given: Sustainable Aquaculture Technologies, how they can help for the improvement of economy of any country, or how they provide the employment for the unemployed, and how they can helping for the conservation of environment. That is the ultimate. Actually, whenever I used to give lecture like this, I keep this slide as the first slide in my all lectures. Do you know the history behind this picture? Do you know the history behind this picture? Anyone?
Protein deficiency, marasmus, and quality of life.
Yes. Actually, this picture was taken by a famous South African photojournalist. He is Mr. Kevin Taylor. See, Mr. Kevin Taylor is a very famous photography journalist. He is from South Africa. This person, when he was visited Sudan in 1993, he took this picture, for which he received Best Photography Award. He received World Best Photography Award. But after receiving the Best Photography Award, he committed suicide. Do you know why he committed suicide? The history of this picture. The Sudanese girl child. See, you know, all undeveloped countries, especially African countries, the malnutrition, poverty, and this is a major issue. So every day in a United Nations camp, UN camp, they serve the noon meal. The Sudanese girl child, she is trying to go to the UN camp where the noon meal is served. Unfortunately, before reaching the destination, she is dying. She is dying. She is almost dying. There is a vulture behind this Sudanese girl child. That vulture is waiting to eat this Sudanese girl child after her death. She died, for which he received Best Photography Award. Instead of taking photo, he helped the Sudanese girl child to go to the UN camp. She may be saved. So why I am telling this whole story? This is the situation in the condition in all undeveloped countries, even in India. We are talking that India is going to be a superpower. India is going to beat China. It never happens. It never happens. Because we are far behind China and other developed countries. Why? Because we are also facing malnutrition and poverty. In India also, we have a lot of poverty, malnutrition. This is a statistical report given by the World Health Organization. There are more than 130 million people across 53 countries in the world. They are facing acute nutrition, malnutrition, and poverty. Similarly, one side food crisis is there, other side water scarcity is there. You know very well what is a major source of any production. Okay? Agriculture, fisheries, aquaculture, the water is a major medium. Without water, we can't produce any food commodity. Okay, so one side we are facing food scarcity, other side we are facing water scarcity. So already we are facing problem for water. Then how we can grow our food crops, agriculture crops, aquaculture, or fisheries food crops? This is the situation. So it is predicted that the next world war is going to come for water. If the next world war comes, that is for only for water. That is so that they are given war for water. There is a slogan. If you go to any website, DSTR website, they are fighting for water crisis. They are providing funding for projects, any project related to water conservation. So that there is a war for water in future. Okay. See, Indian status. Our Prime Minister is keep on telling that, he has keep on speaking, India is. But see, there are 15% of Indian population every night sleeps hungry. 194 million Indians are undernourished. The national shame. This is a national shame. It is stated as national shame. You can see recently the global hunger index is released. Global hunger country index. There are 198 countries in the world. Among the 198 countries, 170 countries have been declared as a hunger country, in which India is ranked at 104. So India is taking place in global hunger country. This issue is not discussed any prime time channel. This issue is not discussed any magazine, any newspaper. Whereas the 9th hour of marriage is discussed a lot. Dhanush divorce is discussed a lot. No channels discuss this issue. Do you know this issue before this? This is our situation. Same time, India is also facing water crisis. We are facing food crisis, one side, other side, water crisis. Employment, unemployment. So this unemployment, food crisis, water crisis, which is related to this economy, environment, and employment of any individuals or any country. So is there any solution to solve these all issues? Is there any solution? Could you please tell the solution to solve this malnutrition, hunger, poverty, unemployment, our environmental threats? Our mother environment is getting polluted every day. Day by day, our mother nature is getting polluted. Our rivers are polluted, seas polluted, land is polluted. Pollution is keep on increasing because population is increased. Human population increased, so that the pollution is also equally increased. Is there any solution to solve this all whatever issue we have discussed? Yes. Ultimately, the agriculture is the only solution which provides the food, employment, and conserve the environment. There is no other sector. We can produce n number of computers. We can launch n number of satellites. But no, of course, we need development also. There is no other. We need both. Economy also we need, development also we need, environment is also important. People also important. We are launching a lot of satellites now. We are bringing AI, artificial intelligence, IoT based system. But the poor is going poor, richer is going richer. So agriculture. Now the India is an agriculture country. India is a country who has a lot of population. They go globally, we are standing first in population. We beat China. So agriculture is our backbone now. But now, due to water problem, water crisis, there is no water for irrigation. Of course, there is no water for drinking also. We are facing for water issues for domestic application, domestic purpose also. So according to the Ministry of Agriculture and Farmers Welfare, they have given lot of data due to failures in agriculture, because of failures in monsoon, because of failure in rain, there are a lot of farmers are committing suicide. It is our Indian status. For our own Indian government, they are putting this data in the Ministry of Agriculture and Farmers Welfare, Agriculture Farmers Welfare. There are lakhs and lakhs of farmers are committing suicide because of failures in their agriculture production. Because there are many livelihoods in agriculture. So no water, then how they can produce their food? Others say the environment is getting polluted. There are a lot of threats for environment. Global warming, climate change, over-exploitation. These are major issues we are facing nowadays. Because of this, there is a report given by the United Nations. If the pollution, climate change, global warming or over-exploitation continue, in 2050, there won't be a single fish in the ocean. If this situation is continued, in 2050, there won't be a single fish in the ocean. You know, the ocean is a major source of fish. If no fish in the ocean, how we can get the fish? Because the fish is the cheapest protein source. It is popularly called as poor man diet. Fish is popularly known as poor man diet. The poor or middleman can able to purchase the fish in a affordable price. Whereas the mutton is around 800 rupees or 900 rupees or 1000 rupees. Whereas the fish is available at 100 rupees. Or sometimes you go to coastal area, one kilogram of fish is 40 rupees. It is very poor man diet and it is protein source. It has all essential nutrients for human being, but fish production is declining nowadays because of this reason. All reason can put a pressure on the fisheries, and the fish production from the ocean is declining, keep on declining. So, if no fish in the ocean, the alternate source of getting fish is culture fisheries. So that we are talking aquaculture. Okay. So China is the topmost fish producing country in the world. China is the topmost fish producer. They are the topmost producers from both in captive fisheries and culture fisheries. In aquaculture also they are top. India they are standing second place. Okay, China they are keeping the trend. They are always keeping the first position in culture fisheries and aquaculture in captive fisheries. Whereas India, now Indonesia, they beat us. Indonesia, they rank second place in culture aquaculture. So this is the situation. So we have aquaculture, the alternate source of fish, but aquaculture is also facing a lot of problems. Disease. There are a lot of disease outbreaks. There are a lot of diseases. As like HIV in human, there is a white spot virus in the shrimp culture. You can see here, this is white spot. This is a white spot disease comes within a fraction of minutes, all shrimp. Like that, there are a lot of diseases. This is a fish tumor. This is a gill rat. See, there are a lot of diseases in culture fisheries, aquaculture. So to cure the disease, the farmers they are putting a lot of chemicals and antibiotics. They are dumping all chemicals and antibiotics available to cure the disease. Of course, these using antibiotics or chemicals will give some partial success. But whatever chemicals or antibiotics we are using in fisheries or aquaculture, they become residual. They become residues to consumers. That chemical they can get go to end users, people. So we will get a lot of health disorders because of these residual chemicals. So the alternate source. Now the using chemicals or antibiotics in aquaculture or fisheries are banned. No more antibiotics or chemicals used in aquaculture or fisheries. Whereas agriculture is permitted, fertilizer or pesticide. In aquaculture and fisheries, the chemicals and antibiotics now banned. No more antibiotics can be permitted. So that now the farmers they are looking for environment friendly, sustainable aquaculture technologies. Without using any chemicals or any antibiotics, even without using pellet feed, artificial feed, a farmer they develop one technology that is called aquaculture, on which we are going to discuss. So we have a lot of sustainable aquaculture practices like agriculture. This is race culture. where you can start the fish larvae and they can grow by using the natural feed. Okay, then see, this is the recirculating aquaculture system. RAS, otherwise called RAS. மறுசுழற்சி முறையில் மீன் வளர்ப்பு. In Tamil it is called மறுசுழற்சி மீன் வளர்ப்பு. The same water can be used regularly, repeatedly, by using some filters. The water from a fish tank go to the filters where all waste materials will be filtered, water purified water will be again come to the fish tank, so fish will grow happily without any disease or without any water quality issue. This is called recirculating aquaculture system, and then this is called closed recirculation aquaculture system. This is high-tech so far. The high developed countries like Taiwan, Vietnam, they are practicing this system. It's a closed recirculating aquaculture system, whereas in India we are not practicing because the product investment is very high. You have to invest, initial investment will be very high. This bioflak, you might be heard about bioflak. Bioflak aquaculture is another environment friendly aquaculture system where you can use the natural feed like microalgae or zooplankton. Only with natural feed you can grow the shrimp or fish along with some probiotics bacteria which will take care of water quality. It's called bioflak. But bioflak also has some disadvantages, so that the IMTA. This is very recent techniques in aquaculture. IMTA. IMTA means integrated multi-tropic aquaculture. Integrated multi-tropic aquaculture, where the fish, shrimps, seaweeds, cucumbers, sea cucumbers, gasteropods, bivalves, mussels, oysters will be combinedly cultured in a confined environment, in a cage. You can put a big cage, and in the big cages can be segregated into compartment, different compartment. In each compartment, one organism will be cultured now. So waste from one organism, as Dr. Kadire said no yesterday. The waste from one organism is a useful for another organism. So here, the fish, shrimp, they can release the waste from fecal matter. That fecal matter will be a useful nutrients for the seaweeds to grow. So the seaweeds, they can utilize that available waste material, the fecal matter or unutilized feed, as a source of nutrients. And the mussels, oysters, they are filter feeders. Whatever any left of dead will be available in the system, that will be consumed with biofilters like mussels and oysters, so that we will get the core product from a single environment. We will get fish, shrimp, okay, oysters, mussels, seaweeds, sea cucumber. This is a very beautiful system. It is developed and it is implemented in most of the Southeast Asian countries except India, because Indians are always very slow. We have enormous resources with us, but we are not producers. We are not a producer. We used to be we want to be a only buyer. We don't want to produce, even though we have enormous site, suitable site, and we have very good resources. But we are sending all our resources to the United States and bringing it from the United States. This is our culture, because we have evolved in such a way. சினிமாவில் சொல்லுவாங்க இல்லையா, செகப்பார் கலரா இருக்கிற போய் சொல்ல மாட்டாங்க இல்லையா, கலரா இருக்கிற போய் சொல்ல மாட்டாங்க. அது மாதிரி நம்ம எப்பவுமே we want to purchase only the imported items. We don't prefer our own native products. So this IMTA is a very beautiful system. Not even a single institute is started in India, whereas in Taiwan, if you go to Taiwan, Thailand, Japan, China, they are producing tons and tons of seafood through this method. Okay, then seaweed, you can see seaweed is a very potential algae. Every day, in day to day, we are using seaweed. You know that today also use seaweed unknowingly. Today also use seaweed. Do you know that? Today use seaweed. Could you please anyone can tell how we use? In toothpaste, seaweed is there as an ingredient. In all the dairy products, starting from ice cream, chocolate, cookies, all bakery products, in all the dairy products. Even the mother Jigadana is there, no? Jigadana, they are using seaweed as a jelly. There are seaweed is a very potential plant. Of course, your correspondent, knowledge correspondent is interested to develop the seaweed farming, algal farming. So that Madam Sumati Madam, she established microalgae culture. Yeah. So seaweed is one of the potential marine plants. You can take any seaweed, you can come out with very novel compounds. That's what of industrial trade. We have gifted with very good seaweed resources. In terms of from Dhanushkodi to Tuticorin, we have 21 islands. In all islands, we have very rich resources of seaweed. So the Tamil Nadu is really gifted with nature. It's a nature gifted state. Then aqua mimicry. I am coming to my own topic. Aqua mimicry. What is mimicry? What is mimicry? We used to there are mimicry artists are there, no?
Imitating someone.
Yes, imitating someone or imitating something. The same technique this is. That aqua mimicry is nothing but how the fish is growing in the sea. Without no one is giving feed, no one is providing oxygen, but the fish is growing happily in the system. The same way, the fish or shrimp, any food commodity will be cultured using this system called aqua mimicry. That is nothing but we going to live with nature without any industrial feed. That is called aqua mimicry. Okay. Last but not least, aquaponics. Now, this aquaponics business has become very popular. Yeah, 36 வயசுல 16 வயசுல முடிச்சு 36 வயசுல யார் நினைச்சது 16 வயசுல யார் நினைச்சது அதெல்லாம் கரெக்டா ஆன்சர் பண்றோம் 16 வயசுல யார் நினைச்சது ஜீரேவி 36 வயசுல யார் நினைச்சது ஜோதி இப்போ நான் உங்களுக்கு அடுத்த கொஸ்டின் கேட்கிறேன் நம்ம தமிழ்நாட்டுல ஒரு சயின்டிஸ்ட் அமெரிக்காவில் நாசா இஸ் a defense ministry of America, US. A person from a very rural village from Tamil Nadu, he is working in NASA and he identified microbes from space. Space வந்து ஒரு microbes identify பண்ணி அந்த microbes டாக்டர் அப்துல் கலாம் பேரை வச்சிருக்காரு. அவர் யாரு? நல்ல கண்டு ஒரு பொலிட்டிக்கல் லீடர் இருந்தார் 2 நாளைக்கு முன்னாடி. அவர் எத்தனை பேருக்கு தெரியும்? எழுதலாம் தெரியும். யங்ஸ்டர்ஸ் அண்ட் மாஸ்டிங் யங்ஸ்டர்ஸ். ஆனா இப்போ ஒரு பேரை சொல்றேன் எல்லாருக்கும் தெரியும். விஜய். இதுதான் நம்ம சரியா? So I don't want to go beyond this limit. Aquaponics. Aquaponics is another nature gifted system where you can grow fish and vegetables in a confined environment without affecting environment. This is a very good system. Madam, you can also propose to your correspondents. You can develop some aquaponics, model aquaponics in your campus. And that is the Nirmala College. They developed a small scale model aquaponics. Okay, madam. Please, madam. We will extend our technical support, no issue. Because we have established commercial scale aquaponics in our university campus. We culture different varieties. It is a very good model system. You can produce our own need, produce our own food. Okay, and using wastewater without using any single chemicals or pesticides. The plant is growing well, excellently they are growing. I will show you the result. So let us very quickly we can go aqua mimicry. What is aqua mimicry? Aqua mimicry, as I said, using natural feed without using a pellet feed or artificial feed. We can grow the shrimp or fish, which is environment friendly. This is invented by a farmer, not by a scientist or academic. That is the beauty. This is aqua mimicry. Very simple definition. It's a revolutionary technology. Okay, it is initiated in Thailand in 2013. A simple farmer developed this technology where he cultured the shrimp using the natural feed like microalgae, copypods, amphipods, and probiotic bacteria. This only these four: microalgae, copepod, algepod, probiotics bacteria. Only using four natural feed, live feed, otherwise called live feed. This shrimp is growing well without any disease outbreak. He never used a single pellet feed. That is very beauty. So yesterday, Dr. Murali Shankar, he said no, 60% of the production cost is going for feed. If you adopt this technology, you can save the 60% of the production cost. The farmer is practicing. You just look at it. This video I am going to give you on the show one video. That is the video. Actually, the farmer is uploaded in the website. You can download it for free in the Google site. Okay. So this technology is really nature, environmental technology. Farmers, they like this because it is economically feasible, environment friendly. It's called aquaculture. Using algae, copepods, and beneficial bacteria or otherwise called probiotic bacteria. Now I am coming to my own expertise. Copepods. I don't know how many of you are doing this. Of course, the zoologists they may be knowing. Copepod. I will give 1 liter of water, sea water. If you analyze, if it is phytoplankton, 80% is microalgae. That is among phytoplankton, 80% is diatoms. If I give 1 liter of sea water, if you analyze that sea water, 80% is composed of copepods. That diatom is a primary producer, or otherwise the microalgae, copepod is a secondary producer, or otherwise the zooplankton. If you remove these both phytoplankton and zooplankton, both diatoms and copepods from the aquatic system, nothing will be there in the system. So we are taking fish from whatever we are consuming. This all only because of this simple tiny cell phytoplankton and tiny animal zooplankton. So this such a phytoplankton and zooplankton is a very wonderful organism which can support the entire biodiversity of aquatic system. So this copepod, actually we are growing the copepod in our department. We have see why we are preparing copepod. We have artemia. You might be heard about artemia. What is artemia? Artemia, rotifer. Yesterday, Dr. Damodaran, he showed some slide. Rotifer, artemia is a live feed, natural feed. If you grow any fish in a pond, there will be a rotifer, there will be a cladocins, Daphnia moina. So this copepod is another important natural feed on which nobody is working because culturing this copepod is very difficult. It is a very potential live feed. It is rich in nutrients, protein, digestive enzymes, amino acids, fatty acids, pigments, minerals, all essential nutrients are there. So that now, instead of eating fish, now the copepod is become a popular human diet. Instead of eating fish, if you eat 1 kilogram of fish, if you eat copepod of 100 gram of copepod can give equal calories of 1 kilogram of fish, eating fish. Such a wonderful organism, copepod, it has lot of advantages. So by using this as a feed, we can grow the fish and stream without any disease outbreak. It has lot of advantages. It is rich in protein, rich in digestive enzymes, essential amino acids, omega-3 fatty acids, antioxidants, vitamins, minerals, all nutrients are there. Normally, yesterday one student he asked one question, no? If you want to increase the nutrients level in live feed, is there any technique? Yes, that technique called enrichment, bio-enrichment. See, artemia and rotifer, nutrition, they are very poor. So that in hatchery, fish or shrimp hatchery, the hatchery producer, they are enriching the rotifer and artemia with commercially available enrichment medium, then feed to their larvae, which is increase the cost of production. Even if you enrich the rotifer and artemia with enrichment medium, it is not equal nutrition level to the naturally cultured copepod. The unenriched copepod contains more nutrients than even enriched rotifer and artemia. So that we developed this copepod in our laboratory. We have studied all characters. Copepod, it has different characters. It has nutritionally superior. It has different movement pattern. It has different characters like pheromone. Do you know what is pheromone? Pheromone type of simply there is smell. Pheromone is smell. It's a type of hormone. Coimbatore it has very good pheromone to induce the fish larvae for easy digestion and bioluminescence. So because of copepods are containing this character, now the copepods become popular natural feed in aquaculture sector, and the farmer they are growing the copepod and they are stocking the larvae, shrimp larvae. By eating the copepod, the shrimp larvae is growing very faster. Within 90 days, they reach the marketable size, harvestable size. Otherwise, 150 days they have to grow. You have to grow the shrimp to reach the marketable size. It should be grown for 150 days. If you use the copepod, the marketable size will reach on 90th day. So that we will save 60 days expense will be saved. So the farmers will, with the minimum investment, they will get more benefit. These are the copepods. It is available. Everywhere. Copepod is available. Wherever water is there, the copepods will be available. So we are collecting the copepods from different ecosystems and isolating, and we culture the copepod and we are maintaining the copepod in our lab. And whenever we need, we can do the mass culture. Okay, this is an example for copepod. And this is a technology we implemented to get the high density for copepod production. By producing the high density copepod, we can culture. This is the population density. Okay, population density. You can see this. Please click on this slide. It's a video. Not okay. So it's a video actually. We culture the copepods in a 5-ton, 10-ton tanks. We get very good biomass. That biomass can be harvested and given feed to the fish or shrimp larvae, and we analyze all parameters. Next slide. Otherwise, I will have a separate video. Next slide. So these are some videos I showed you. We have separate videos, right? Next slide. Okay, this is actually RA facility, as I said, recirculating aquaculture system facility. We have established our university campus with the financial support of DBT, in which we culture in the copepods, microalgae, and we also have the facility to do the experiment, fish experiment, larval experiment. We have all facility in our university campus. Next slide. And we analyzed all the parameters, growth parameters. So we culture the copepod. We use the copepod as a feed for the fish and the shrimp and both edible fish and ornamental fish. In comparison with the traditional fish, controlled fish like rotifer and artemia, we analyzed all growth parameters. So these parameters, so you can see here, this is sea bass, kodua meat சொல்லுவாங்க இல்லையா, இது வந்து கடல் கொடுவா. கொடுவா மீட் வந்து ரொம்ப டேஸ்டா இருக்கும். இது வந்து carnivorous fish. You can see here, this is the length and weight of the fish which grown in different feed like rotifer, artemia, and copepod. The copepod fed fish always showing very good growth and also survival compared to rotifer and artemia. This is the result I have given. Next slide. This photo you can see here. This is the same size fish stock. The same stocking size, same duration, 20 days duration. The rotifer fed fish, they can grow only this size. The rotifer grown fish larvae, they can grow up to this size. Whereas the artemia normally grow this, and this is the copepod grown fish larvae. So copepod can increase the growth of the larvae within a stipulated time 10 folds higher growth than the rotifer and artemia. And This is actually one ornamental fish. You can see here, we used different fish, Artemia and Coimbatore. This is lithography fish. And we have got very surprising result in ornamental fish. Coimbatore is not only improving the growth or survival, it is also improving color, pigments. You can see here the pigments. This is Artemia filled fish. This is Coimbatore fish. You can see the color improvements in the Coimbatore fish has lot of improvements. Body pigmentation in the caudal ciliate, the entire body, dorsal fins, whereas the Artemia filled is less Coimbatore. So it shows Coimbatore is not only improving the growth and survival, it is also increasing the pigment. So that if you use Coimbatore for ornamental fish production, you will get more pigmented ornamental fish, and if your fish has more color, the rate will be very high, price will be very high. So that you will get more income. Okay. And this is the marine fish. You can see we use a different combination. Coimbatore alone. This is 50% Coimbatore and 50% rotifer. And the same duration, same density. On 14th day, the Coimbatore fish has grown very well, whereas the 50% Coimbatore and 50% rotifer compared to less growth. So this is marine fish. Okay. And we also tested with the shrimp. You can see here. And this is the shrimp larvae, Pinus monoda, the same starting size, same duration, 21 days. This larvae which grown on rotifer, this is the animal which grown on Artemia. This is the same animal which grown on rotifer. You can see here there are lot of pigment improvements in the entire body. This pigment, we analyzed what is the pigment, that is astaxanthin. This astaxanthin, actually we have reviewed the literature. If you consume more astaxanthin in your diet, it improves your immune system. So if you have good immune system, you can escape from most of the pathogenic diseases. That is the report given by the scientists. Okay. And we also tested with other. This is another shrimp, Pinus venati, is American shrimp. Okay. We tested with different feed, and Coimbatore shows very good growth. And this is the video now I have told you. A farmer is doing. You can see, you just carefully listen to this video. This system is aquamimicry, or otherwise called coffee technology. With Coimbatore and Aquaculture, he started his culture, shrimp culture. You can see this video. This is a Coimbatore, it's biomass. It is available in the website. You just put the keyword aquamimicry or coffee technology, you will get this video from the Google site. So actually this farmer, he started 9,000 fry in 0.4 hectare, not in 1 hectare actually. Normally in India, we use 1 hectare pond for shrimp culture. But here, he used only 0.4 hectare pond, started with Coimbatore, Aquaculture, microalgae and probiotic bacteria. And the larvae will be stocked directly without any adaptation or acclimatization. And they grown for some certain period. You can see the summary of this video. At the end, you can see the summary. This is DOC. DOC means days of culture. 21 days culture size. This is the shrimp which grown for 21 days. So every week they used to take sampling. Yesterday, Dr. Kadres said, no, sampling we have to do. Every week we have to do the sampling to check whether the animal is taking feed properly or whether the animal is growing properly. And they are using different, what is called, aerators. This is patented aerators. Okay, this is a lined pond. Lined pond means the earthen pond will be covered with polythene sheet so that we can avoid some diseases. And this long patented aerators use. And this is bottom aeration. To support the oxygen to Coimbatore or Aquaculture, we have to provide oxygen from the bottom because it is the bottom living animals. And they are making sampling for every day. Actually, they feed 330 kg per day. Per day, 330 kg of live feed. Coimbatore will be utilized by the animals. The average body weight 8.33. Average body weight 8.33 gram at 120 cows. For aquaculture experts, it is very easy to understand. So this is you can see how animals is very neat and clean. Whereas in traditional farming system, if you look at the animals, they are very dull. They are very dirty. Okay, but you can see that this is black body. This is very important. the fluid volume is very less. The fluid volume is nothing but turbidity. If water turbidity is high, it will choke in the gills. Okay, the animal they will not be able to take the oxygen, so that the animal will become a stress condition and easily the disease will come. Whereas here, this is DOC, 60 days, not even 90 days. That farmer, he cultured for 60 days and he got a total production of 10 ton in 1.4 hectare of pond. This is the way how we are doing sampling, weekly sampling. So average body weight on 67th day, 12.19 gram, and 1 kilogram 82 pounds of animal. 1 kilogram of means it has 82 individuals. So they are harvesting the product, biomass. At the end of the 67th day, they harvested 9,260 kilogram, 9,009 ton, 9.2 ton product in just 0.4 hectare area. The benefit, the production summary, 1 kilogram. In India, 1 kilogram 500 rupees. You just calculate 9,000 into 400 or 500. How much it become? 9,000 into 500. You put 400 or 500. See, this is the production summary. The total biomass 9,260, duration 67 days. FCR, you can see that FCR. FCR means food conversion ratio 1.1:1. To grow 1 kilogram body weight, to reach 1 kilogram of weight, or to 1 kilogram of biomass, the animal consumed 1.1 kilogram of feed. It's very good FCR. So the FCR should be less, then only the farmers will get benefit, more profit. If the FCR is 4, consuming 2 kilogram feed and grown only 1 gram, 1 kilogram means it is a failure. It's a loss, production loss. Whereas here you can see 1:1, 1.1:1. That is very good sustainable technology, and the total biomass we harvested around 9.2 ton. 1 kilogram 500 rupees. We calculate how much we got at the end of the 67th day. This is called what is called the aquaculture. என்ன சொல்லுவாங்கன்னா aqua sea gold அப்படி சொல்லுவாங்க. Or otherwise blue dye. Because shrimp has lot of price value. So until 1990, India is a major shrimp exporter. But because of the disease outbreak, now actually we replaced it by nature. So this video is available on the website. If you are interested, you can just go Google it and you get this video. So this is for aqua mimicry. So without using a single pellet feed, without using any pesticides or any antibiotics or chemicals, we will produce fully organic product and it is environment friendly because this system is not affecting any environmental issue. The second one is aquaponics. So aquaponics, very quickly let me go. As I said, the aquaponics very nature filter system with a very simple facility, you can do wonders. If you have any damage or partially broken any tank or any container, you can use that container to grow the fish. And another container for growing vegetables or plants. So this is nothing but the method of growing agricultural crabs and fish together in a recirculating system. This is a simple recirculating system. Let me explain. We are growing the fish in a tank. That fish culture tank has lot of waste. The source of waste is fish excreta, fish fecal matter, and any leftover feed is that is a source of nutrients. That is rich source of nitrogen, ammonia. Okay, that waste water can send to the plant beds where the vegetables or plants will be growing. In the plant bed, there is a microbe. Nitrosomonas nitrifying bacteria, which will convert all this organic waste into inorganic nutrients, nitrate, phosphates together. That nutrients will be consumed by the plants will grow. Once the water leaves the plant bed, that water again, filtered water comes to the fish tank. Because sand or pebbles is a very good filtering system. Very simple system. Very traditional system. நம்ம முன்னாடி என்ன சொல்லுவாங்கன்னாக்கா ஒருங்கிணைந்த மீன் வளர்ப்பா, மறுசுழற்சி முறையில் ஒருங்கிணைந்த ஆர்கானிக் ஃபார்மிங் சொல்லுவோம் இல்லையா, integrated farming. The same integrated farming, but there are some sciences involved here. The science is nothing but microbes. So instead of microbes, we can use other organisms like worms also. So these are the advantages of aquaponics. Why we are recommending aquaponics to go for aquaponics? The reason is it requires less than 2% of water that can be used in traditional farming. Traditional farming, we need voluminous water. We have to tilling, we have to weeding, all the practice we have to do. We have to use fertilizer, pesticide. Whereas here, no fertilizer, no pesticide. Moreover, the water, it requires only less than 2% of the water when compared to traditional agriculture. Okay, the water is recirculated. Here the water is recirculated and we will get two crops, one is fish, other one is vegetables. And this is a nutritious food also. Moreover, here we are not using any chemicals, any pesticides. And this aquaponics is very famous in Kerala. If you go to Kerala, there is one hobby there. They are doing aquaponics. Kerala is a state who is implementing all the any sustainable aquaculture technology coming into the practice. Indiaவிலே ரெண்டு பேர் ரொம்ப பிரில்லியண்டான people. Two state people. Last two state. I will ask this question. All time, any wherever I used to give this sector. Two questions I have in my mind. Who is very active in the world? Very active person. Yara Sona. Very active person in the world. Among the 24 hours, 18 hours they used to work. Very active. Japan. India, the state people, they are very brilliant. The two state people, two state people. One is Kerala, other Tamil Nadu. Ilya, Tamil Nadu is very good. Kerala is correct. So other state was West Bengal. It's both Kerala and West Bengal. If you look at Indian history, political history, the highest position occupied by these two people. Two states, West Bengal and Kerala. Why? Because these two state people, they are fish eaters. They eat fish. In Kerala, daily we do lunch. If you go to the West Bengal, the fish is classified in vegetarians. Fish vegetarian. So why I am telling this? Fish is provide all health benefits to the consumers. And அதே மாதிரி ஜப்பான்காரங்க கடல்ல இருக்கிற எல்லாத்தையும் சாப்பிடுவாங்க. You never leave any even sea snake also you used to eat. So that the seafood protein keep you mentally and physically healthy. So that Japanese they are very active people. They used to work very actively. So because of these advantages, the aquaponics now is become popular. This is the secret behind the aquaponics. You can see. Okay, here the bacteria is playing a major role. Fish produce waste in the form of ammonia. The available microbes we convert it into nitrates. It will be utilized by the plant, vegetables. Again from the plant bed, the water comes to the fish tank. It is a simple recirculating system where you can grow the fish and vegetables at a time in the confined environments. So instead of microbes, you can also use worms. There are worms is there now, which will use all organic waste converted into inorganic nutrients. Okay, and this is the system different components in aquaponics system. So we need, if you go for commercial scale aquaponics, we have to have some machineries. Machineries like filtering system, okay, for commercial production, large scale production. And this is a different aquaponics technique. There are three techniques: NMD, nutrient film technique, DWC, deep water culture system, and media bed system. So using these three techniques, we can grow the vegetables in a system, hydroponics, and fish in the aquaponics. Okay. So this is, you can see nutrient film technique. You can see in our lab, we established lab scale. Here in the tank, we are growing the fish. The water from the fish tank will be pumped using the submersible pump. The water will be, okay, flow through here in and the tube, simple PVC pipe. In a PVC pipe, we are growing some plants. You can see that this is green crab, pacifier. So in the commercial, we can grow different crabs, and this is in a commercial scale. Beetroot. They are growing beetroot in NFT. If you go to Bangalore, you just Google it, Madhavi Aquaculture in Bangalore. Madhavi Aquaculture farm. They are growing vegetables, fish. They are exporting every day. Madhavi Aquaculture farm. It is located in Bangalore. This is a commercial scale. Okay. And this is a deep water culture in which we can grow the water plant. For example, wind. We can grow the wind. Okay. So this is a commercial scale deep WC system, and the media bed system. Okay. So in which instead of sand or soil, here we can use the pebbles, gravels, or hydrochlore, clay pots. The clay pots are otherwise called hydrochlore. It is repeatedly we can use. One crab is over, we can wash that pebbles or hydrochlore. We can use for another crab. Here no tilling, no tracting, no weed, no chemicals, no pesticides. Okay, and the plant is growing there. See, this is commercial scale media bed system, and we experimented different plants and different fish. This is using different media bed system. Okay, and this is a deep water culture system. This is NFT system. We culture different animals. Due to time constraint, let me go fast. This is a fish, one ornamental fish, Oceania spinos. We culture in control system. Control system is a traditional system. And you can see the difference. This is a fish which cultured in the control system. This is NFT, aquaponics, and media. You can see how it is big, how it is larger in size. Okay. And we also cultured the other. See here, very important only is the water quality. You can see the water quality. The dissolved oxygen level in control was 5.98, whereas in aquaponics system 6.4. And you can see the ammonia and nitrate level, how it is reducing in aquaponics system. See, this is actually ammonia. Ammonia, it was 0.09 on 15th day, whereas in aquaponics system 0.03 and 0.01. So all available ammonia are nitrate in the waste converted into nitrate, so that the plants will consume and we will get good products and the environment friendly. This system is not affecting environment. It is purely environment friendly system. And see, this is what is called possibility. How you can see how it is growing luxuriantly in the system. Okay. And this is a tilapia. Tilapia is the best fish to culture in aquaponics system. You can try your college also. You can see this variation in size. See, this is a control grown fish. This is a aquaponics cultured fish. Okay. And this shrimp also we tested. Today's phenomenon. Okay. So shrimp also growing well in the aquaponics system compared to this traditional system. This is a sea bass. I don't know sea bass. You can see the sea bass how it is bigger in aquaponics system. This is a control system. Same stocking size, same duration. The aquaponics results very promising when compared to traditional system. Okay, this is a tomato. Actually, tomato is one of the vegetable which is growing very luxuriantly in aquaponics system. Even the tomato and the beetroot we can culture in salt water also. Up to 550 salinity we can culture these two crabs. And this tilapia again. And this is the system we have established in our university. This is Tamil Nadu government. They funded our projects 67 lakhs constantly, and we established this system. Okay. See, the video is not working. Or else no problem. I will show now. Separately, I have a video for you. And this is the facility we have established in our university campus. So, in time permits, you please visit this facility. Actually, so far we have cultured tilapia, sea bass, venami, and tomato, chili, green chili, mint, even small onion. Grapes also growing well in aquaponics system. We tried. This is another video. This is the media bed system using the gravel. This tomato is growing, and I show the. This is the fish which cultured in the aquaponics system. Your system, your laptop is supporting for this, right? Anyway, I will operate it separately. Okay. And we culture seaweed also. In aquaponics, we culture seaweed, we culture salt marsh also, Salicornia. This all tomato. We cultured tomato. Tomato actually we analyze the phytochemical and biochemical. Taste wise, the aquaponics, I mean hydroponics cultured tomato has very delicious taste than the traditional cultured one. So there is one woman scientist working under me. She is a botanist, so she is taking care of all these. We cultured beetroot, okay, and this is production summary. This shrimp and see the growth in the aquaponics system. Okay. This is actually salt marsh. Salicornia plants. It is rich in iodine. Coastal area, the plant is very common. It is edible, human edible. Either used for pickle, they prefer pickle, salt marsh pickle, Salicornia pickle. If you taste it, it's very salty. Okay, and this is edible. They prefer the pickle and the vegetable salt marsh. They prefer. So these are the crabs. Not all the crabs are growing suitable to grow in aquaponics. Not all the plants or not all the vegetables can be able to grow. Only some particular species, for example, tomato, peppers, cucumbers, broccoli, and greens, beans, beetroot. Okay, these are the identified crabs we can grow in aquaponics system. Likewise, in fish also, not all the fishes can be cultured. Only some varieties. For example, tilapia. Tilapia is the best species to grow in aquaponics, to my knowledge is concerned, followed by sea bass. Okay, that too you can take gift variety. Gift tilapia is very delicious. You can try it. I don't know whether it is available in Coimbatore. Gift is very delicious fish. It is available nowadays. It is a cultured variety. Okay, so that you can use tilapia, trout, sea bass, and koi carp. This is a carp, koi carp. Koi carp is ornamental fish. And the shrimp, the sea bass, shrimp both the Damodaran and Damodaran we can culture. Okay. So we conducted several training programs and awareness to celebrate women and school students, even college students, to create awareness among the school and college students to become an entrepreneur. Because this is one of the best entrepreneurship valuable system. Okay, aquaponics. And some scientists in this video actually, as I said, no, you can just Google it, modeling aquaponics. You will get that video. So as the aquaponics is environment friendly, it is economically viable. Anyone can do, even in your terrace, home terrace, as like horticulture, you can do, and you can try for your home, and you can establish this small scale terrace aquaponics system in your office also, in your college, as a mini project, as a project, you can do this aquaponics. You will get a very promising result, fruitful result. So with this, so this is very important. Like, so I have given the title, the sustainable aquaculture technologies, how it is supporting the economy, environment, employment. You can see here, there are 179 billion tons value of fish with the worth of $404 billion. Every year, we are getting this much economy through the fisheries and aquaculture, and we contribute 1.5 trillion annually in value-added products. That $401 billion through fisheries and aquaculture, through value-added products, fish value-added products, we will be getting 1.5 trillion annually. So it is very good economy through fisheries and aquaculture. We are getting a lot of economics, and you can see that through aquaculture, 22.3 million tons, that is inland fisheries. This is very important. I don't know how many of you are doing this. Aquaculture fisheries, they release less greenhouse gas. You can see here in this slide. You can compare this result. Aquaculture and inland fisheries, they release 22.3 million tons of greenhouse gas. Inland fisheries 43 million tons, whereas agriculture 9.3 billion tons. And the meat, red meat 0.83 billion tons. What the FAO released, consume fish, the oxygen, carbon dioxide emission will be very less. And both production and consumption of fish, it will release the less carbon than the consumption and production of agriculture food crops. So it is environment friendly, conserving the environment, and it provides employment. You can see here around 60 million people they get employed worldwide in fishing, both culture fisheries, capture fisheries. So you increase the production, aquaculture production by implementing these sustainable aquaculture technologies like aquaponics and aquamimicry. We can generate the income, we will provide the employment, moreover we will conserve our mother nature because it is environment. So with this, let me conclude my talk. If you have any clarification, you are most welcome. Very important section is acknowledgement. Okay, so I tried several ways to get the photo of our Professor Sumati Man and other faculty members. So my suggestion, of course, I will give my suggestion to our correspondent also. Because our faculty profile should be there in the college. Because nowadays NAC and NIR ranking is there. They are verifying our profile, college profile, by verifying faculty profiles also. So I tried this many times, but I failed to get the photographs of faculty members. So it is the right time to thank the authorities of this esteemed college, particularly the correspondent, secretary, principal, the HOD madam, Sumati madam, who is very active, young and energetic professor, academician, other faculty members, research scholars, students, and last but not least, all participants. So if you have any doubt, I am very happy to answer. So this is a team actually. We have a constant project now. Dr. Raja Kumar, Dr. Rajaram, Dr. Prashanti Devi. These are the two guys working with me. She is a woman scientist, he is a woman scientist. She is Nandini Devi, DPP, research associate. They are the pillars, main pillars behind this establishment of this aquaponics under the Tomsky scheme. So I thank them. I should thank our own university for granting permission. Thank you very much. If you have any queries, I am very happy to answer.
Thank you for your inspiring talk, sir. So you told coping boards are difficult to culture. So why it is difficult and whether it will survive or multiply by its own natural environment, or we need to only culture because you were telling the feed for you want to put for a 1 kg, 1 point something, you have to put. Why it is difficult and why it is not?
No, why it is difficult? See, nature is given in such a way. If some aspects they are good, some other aspects they are bad. See, cope has a lot of advantages. The only disadvantage is getting high density in culture system. Okay, it's very difficult. The reason is it has some inhibiting hormone, which inhibit the less egg production. I mean, which stop induce the less production. That hormones can be knocked out. So that we wait for selective breeding. We wait selective breeding and induced breeding. By inducing the copepod, by giving some external factors like hormones and probiotic bacteria, we induce that copepods to go for high production. So that is the only disadvantage with the copepod. We can't get high density, high production in cultural system. Whereas in nature, it is always very productive because we can't mimic the nature. Okay, if you bring the system into the controlled condition, there are some lag. So the copepod they have some inhibiting hormone, reproductive hormone, which control the egg production in the copepod so that you cannot be able to get the more density. And if you identify that genes which is responsible for that induction, that will be clarified. That is the only issue with the copepod. Otherwise, copepod we can use as even not only fish diet, it is human diet also.
Sir, I have visited your copepod.
Yes.
You have very high lack of copepods. When it is coming to the farmer scale, is it feasible for the farmers to do so? This is the first question. The second question is, the copepods are growing very well in the salt water.
Yes.
But when we are going for inland fishing, is this technique is feasible for inland fishing also?
Yes. Yes, madam. Both the questions are very important and interesting questions. So why the copepods are coming into the market? The reason is, as I said, getting high density. Still, we are trying. Somewhere we come out with some selective breeding, which supports for high density, but it is not supporting our requirement. Because we are producing millions of larvae. To feed the millions of larvae, we have to at least produce billions of copepods. So, producing billions of copepods is a major challenge. The reason is there is some genetic issue with the copepod. So that if we come out with some genetic tool which can trigger the copepod for high production, that copepod will soon come to the commercial scale. So, of course, up to lab level, we succeeded. Lab level up to 5 10 ton capacity, we succeeded by implementing selectivity. If you take it to feed, there will be some challenges. Because copepod is another problem. Copepod is a vector. Copepod is a virus vector. If you take this copepod to feed, then you get the virus, it will transmit to the organs. But fortunately, none of the copepods so far reported vector in the organs. Whereas another temperate waters is reported. So the main challenge is we develop high density production technology, then only the copepods will come into the practice. Now the farmers, they are coming forward, they are getting space from us and they are asking the consultancy also. We are giving some consultancy, technical advice, and one farm in Andhra Pradesh, they already established the tank based copepod culture. We are giving the technical guidance, we are extending to the farmers. So very soon the copepods will become popular in the life field, which will replace the Artemian copepod. That is the only challenge. The another question for marine, copepod is there everywhere. Even freshwater, copepod is there. But the only thing, the marine copepods are highly nutritious than freshwater. The size wise, freshwater copepod is bigger. Whereas the nutrition point of view, the marine copepod has more protein and omega-3 fatty acids than the freshwater. And the density, population, diversity is less in freshwater. Whereas in the marine system, we have n number of fish. So far, 4,500 species are reported globally. In India, we have reported 700 species of copepod. When compared to freshwater, the marine has more diversity than the freshwater. That is the two issues there. But we can culture the freshwater also. We also try freshwater, we develop freshwater copepod. If anyone of you has interest in work on freshwater copepod, you most welcome. We will give training course. The technique is same. To culture the freshwater copepod, technique is same. Only is the salinity is same. Instead of freshwater, we can use seawater. That's all. And based on the salinity, the other parameters can be made. Otherwise, the marine copepod is always better. One species actually, that species can grow in both freshwater and seawater. That is the potential techniques. Yes, that we tested with other alternate also, but microalgae is best to culture the copepod. Microalgae is the best. We also tried with some other herbs also, some plant extract, which one plant will give some promising result, or which actually they are looking for some patent, so that I don't want to disclose. One particular plant, terrestrial plant, it will use the copepod reproduction. But to my knowledge, algae is best, microalgae is best. Yes, sir. Yes, that's it. Master facility we have in our university. So to culture the copepod, first we should start microalgae. Without microalgae, we can't grow. So microalgae is suitable feed for the copepod.
Sir, if we put the copepod in the environment, it will multiply itself, sir. Again, we have to feed every time.
No need to for repetitive stocking. It is a sexual reproduction. It won't multiply. When they get favorable condition, okay, they can reproduce. There is a seasonal breeder. It's a seasonal breed. And during unfavorable condition, that cyst will be stored. Copepod release eggs as we got. the eggs will be in the resting condition, dormant condition. When they get favorable condition, they can hatch.
So, through that shrimp culture,
Yes.
there's coping boards.
Yes.
And I think that would be the microalgae also for
No, if we only one time it will inoculate microalgae and copepods, it will not multiply, they can reproduce.
So one
No need to
time
start.
investment of microalgae, coping boards and shrimp.
Yes.
And
Periodically we have to inoculate the probiotic spectrum. That we have to. Microalgae will come, no need to reintroduce.
what kind of probiotic bacteria?
Bacillus. Bacillus subtilis is giving promising result. Bacillus subtilis.
Sir,
Yes.
copepod increase the pigmentation of ornamental fishes, sir.
Yes.
What about in human beings, sir?
Yes, of course. See, there is one article during Corona COVID time. There is one article published in Aquaculture. What that conclusion of that article. If you consume any food commodity which has more pigments, particularly astaxanthin and beta carotene. If you take more beta carotene and astaxanthin rich food in your diet, daily diet, you won't get COVID. This is the result published by a scientist. It is proved scientifically also. So what they suggest that astaxanthin and beta carotene improve our immunity. It's a very good antioxidant precursor. So that we will get very good immune system. We will escape from most of the diseases. So if you consume copepod, even RK, spirulina, you will get more pigments that astaxanthin beta carotene. So that it will increase your resistance. So that the carrot is known for beta carotene. With that microalgae, we have one microalgae, Donaliella salina. That Donaliella salina has 10-fold higher beta carotene than the carrot. So like that copepod also. Both this microalgae and copepod, it has become popular human diet. If you go to Norway, there is one company. They produce marine oil. That marine oil they are extracting from the copepod, not from the fish. We know the fish oil capsules. Now the marine oil is extracted from the copepod, which has more omega-3 fatty acid and astaxanthin than the fish. So that it is named as ruby oil. You just put in Google ruby oil. What is ruby oil? Ruby oil is nothing but the oil extracted from the copepod, which is a good source of omega-3 fatty acid and astaxanthin pigment. It is very good in human diet. It is a very good supplement, nutrition supplement. Yes.
Sir, you said copepods have inhibiting hormones. If the fishes keep on feeding them, does it affect the fish production, sir?
Really very happy. In one college from Kadalur, a government college, one girl, she asked one question. Sir, you are telling eat fish, it has lot of benefits, but the fish has lot of metal contaminants. If you consume metal, you will get lot of health problem. How do you identify it? Is there any technique to identify whether the fish contains metal contamination? Then I got some idea. Why don't we develop some sensor as like thermometers to check the temperature, body temperature? Why don't we develop some sensor which can detect the metal level in fish? The same way he has. I appreciate you. Where are you from?
ABB College of Tirupur, sir.
Very good. I appreciate you because yesterday also madam asked, you are using lot of hormones. If the hormones use continuously, whether it reach the consumers. Every system, it has the uniqueness. See, the virus, I don't know, still. That virus, it is pathogenic to only that stream. If we consume that stream which has that virus cannot be a pathogen to humans. So, for example, metal is poison. Of course, the Raja is an expert in metal poison. Metals, they won't deteriorate. Whatever accumulate, it will transfer to the next property. Whereas what is hormone? Hormone of course, Professor Manohar is a biochemist man. You know very well more than me. There are some compounds they won't what is called yes, it won't transfer to the other system. That hormone or compound they become inefficient in the particular system. Such a way to induce the which we are using some hormone. That hormone available residues may not become an issue to consumers. But if you go beyond that, there are issues. So it won't create any issue so far. Because we are standardized the dose for hormones, everything, we are standardized. That standardized dose, no issue. If you go beyond that dose, they may create some disorders.
Hormones in the testes are very different.
Yes.
So that hormones are proteins. We don't have that receptor. So
Yes.
that hormones don't have.
Any more questions? Okay, thank you very much for your kind listening. So once again, I thank the authorities, especially Madam, Madam, our frequent visit from Biotechnology University. So I appreciate both of you because they came to our lab, they trained themselves in microagriculture. They Coimbatore far away from sea, even Trichy also far away. In their marine microagriculture, the interest of the day definitely you will start to become one of the output for this area. Thank you, madam, for the output.
Sir, I have a question.
Yes, good afternoon, sir. Good afternoon.
Sir, as you said that you are working marine reports, right, sir?
Yes.
I have a question. There is two types of filters commercially used as a feed for shrimp, mostly in an active state. One is Artemia and another one is Coimbatore. Both Artemia and Coimbatore is also a viral transmitter vectors, right, sir?
Yes.
So, Artemia, sir, Artemia mostly marine use for the purpose use for as a feed. So, to present is shrimp farming. So, Coimbatore from the vice syndrome, viral disease. Attack AIDS how will be identified எப்படி தெரியும் நமக்கு ஏன்னா நமக்கு தெரியும் வைரஸ் சிண்ட்ரோம்ங்கற வைரஸ் வந்து கோபிகோஸ் வந்து வேல்யூ அஃபெக்ட் பண்ணாது பட் ஸ்டேட் வேர் as a vector but it do automatically copico's production right sir
No, actually no virus syndrome so far reported in Coimbatore, but some tumor-like anomalies are reported in Coimbatore. That is an environmental disorder. If environment, the water quality is bad, there are some tumor-like structure. The animal will get some tumor-like structure. That is not virus. That is the disease scientists are telling that it is environmental disorders. None of the reports reported as a viral disease. It is a vector. I agree. Okay. To identify whether the Coimbatore has virus or not, we have to go for a PCR test. As like a testing in hatchery, in a strip you are testing for PCR.
yes sir
Like that, Coimbatore also can test for PCR.
yes sir but அதான் சார் இப்போ கோபிகோட வைரல் டிசீஸ் பண்ணாப்போ நெக்ஸ்ட் ஸ்டெப் சார் ஒயிட் பர்ஸ் ப்ராப்ளம்ஸ்னா ஒயிட் ஸ்போர்ட்ஸ் தெரியும் சிம்பிளா வைரல் டிசீஸ் வேற ஸ்போர்ட்ஸ் தெரியும் பட் கோபிகோஸ் மைக்ரோஸ்கோபிக்கா இருக்கிறத தாண்டி ஸ்போர்ட்ஸ்னால பாட்டா இருக்காங்க
No, yeah, for that we have some staining procedure. There are some high-grade staining, some staining procedures there. You put that stain under microscope, it is visible. Only some virus, some symptoms. But as a Coimbatore itself is a micro animals, we have to go for some cell images, some using some scanning electron microscope, we can detect that spot-like appearance. Okay, otherwise further we can go for the PCR test. This is the available techniques. Yes.
Shrimp culture பண்ணிட்டு இருக்கிற ஃபார்மர் using the technique what as a feed aquaculture right sir அந்த மாதிரி பண்ணிட்டு இருக்கும்போது the shrimp is attacked with some other disease or virus viral disease or some outbreak is in such cases we normally follow use disinfectant and other remedies we use to treat the shrimp If we using disinfectants other chemicals, it will affect the zooplankton's growth, right? So
Yes.
it will cause death in zooplankton, such as Coimbatore, or other phytoplanktons.
Yes.
In these cases, whether you are said previously, will be not a failure, but it will be like a collapse one. For example, if you're using a remedy for the shrimp viral attack, and the zooplanktons were used in their pond will start to spontaneously replacing the zooplanktons growth.
Yeah, both will affect. Or they use the lined pond. In this video, aquaculture, they use lined pond, where no animals contact with any what is it, earthen pond. So the earthen pond is a major source of contaminants. Moreover, here we are not going to use any pellet feed. Okay, the pellet feed actually become a major source of ammonia. That any leftover feed is there, that decompose and they become ammonia. Level will be increased. That animal will become stress condition when no water quality is maintained well. If water quality is maintained well, no issue. So that the aquaculturists they used to tell good quality feed, good quality seed, good water quality management. If you practice these three, there will be no risks. So unless otherwise if you are not maintaining the system in good, healthy condition, there will be any disease. Otherwise the disease will come. And there are some sorts of aerosol pathogens are there, but it is not reported so far. So that they are putting some bad bird pencil. Yesterday, Dr. Kadir's is speak on bird pencil. Bird is a carrier of some pathogenic streams from some other compounds. They can put here, it will be respiratory. But this aligned pond with only natural feed, if you maintain proper water, see, if you use natural feed, the water quality will be very good. Okay, that is natural feed. If any leftover feed is there, that will be utilized next day by animal. So that you don't create any water quality. So that they are not using any antibiotics or anything, any chemicals in the system. So we have to cope up with the natural system so that they will be. So the source of pathogen is a man, human. Anthropological is a major source of pathogen. So if more pollution is there, there will be any disease of it. That is my answer for your question. Are you satisfied?
Yes, sir.
If you have any more clarification?
I can give you that.
Okay. Thank you.
Thank you, sir, for the informative session. We sincerely express our heartfelt gratitude for your inspiring speech on the importance of science in every aspect of our daily lives, and we are truly fortunate to hear this insightful words from you on a very special occasion, National Science Day. We wish everyone a very happy National Science Day. Thank you, sir. We shall now have a short tea break. I kindly request all the participants to assemble back within 15 minutes. As we are running short of time, please assemble back within 15 minutes so that we can start the next technical sessions. Thank you.
Excuse me.
மேம் ஐஸ்வர்யா மேம் லூப்ல போட்டு அந்த வீடியோ
இது வந்துட்டு
participants to assemble faster. The speaker has arrived. Welcome back, all of you. Let's begin the technical session 5. For this, I now request Dr. B. Kalekandi, Assistant Professor, School of Biotechnology, to introduce the speaker of technical session 5, Dr. Damodaran, Professor and Head, Department of Marine Science, Bachelor's University, Coimbatore.
Good morning. Sorry, good afternoon. Please, maintain silence. Those who are standing outside, please get in. Good afternoon, Honorable person here. It's my great pleasure to introduce our distinguished guest for today's national seminar, Dr. N. Damodaran, Professor and Head, Department of Marine Sciences, Damodaran University, Tiruchirappalli, Tamil Nadu. Professor Dr. N. Damodaran is an accomplished academician and researcher with over 25 years of teaching experience since 1995 and nearly 3 decades of research expertise. His academic journey includes advanced degree in biochemistry and botany, reflecting his strong foundation in life science and marine sciences research. Throughout his distinguished career, he has served in various academic positions, progressing from assistant professor to professor, and has made significant contributions to teaching, research, and academic administration. His area of research are very diverse, impactful, including biochemical studies of marine resources, marine actinomyces, and bioactive compound production, microbial studies on multi-resistant pathogens, biomedical applications such as biofilm control, anti-cancer, wound healing, and anti-diabetic research. Dr. Manoharan has guided n number of PhD scholars, MPhil, as well as postgraduate research scholars, shaping the next generation of science. He has an impressive publication record in reputed national and international journals with a strong citation index and significant academic impact. His work on marine bioactive compounds, antimicrobial resistance, and nanobiotechnology has contributed meaningfully to both environmental and biomedical sciences. In addition to his academic achievements, he has actively contributed to institutional development through various roles such as examiner, coordinator, mentor for skill development and student welfare initiatives. Today, we are truly honored to have such an eminent scientist and inspiring academician with us to share his knowledge and insights. I welcome you, sir.
So good afternoon to all. So last 2 days you are very engaged with the aquaculture. So I am not a specialist in this aquaculture, but I am the byproduct of aquaculture. In the session of this, I am byproduct for the development of new novel products. Okay. So this is for the I am inconvenient for the constraints of the times. I am able to just introduction to that some the nutraceuticals or cosmeceuticals or the bioactive compounds through the aquaculture. Okay. So what is the main the objectives or the mode of the objective of this conference? So what is the ultimate for this workshop? Any idea to this? Ultimate aim. You are the biotechnologist, most of the people are marine science or aquaculture or life sciences. That's no any persons from the non-living sources, especially for the engineering or mechanical or we are in the biological system. So what is the ultimate for this workshop? Very entrepreneurship, the aquaculture, and next, you are in the mindful person. That is the point of it. Always that Tirupur and the Coimbatore, they have to think about that 5 districts in the Tamil Nadu. That is especially for the western part of the Tamil Nadu, always thinking about the business. I am coming this point for this the entrepreneur development. So then ultimately we have to then go for the final product of the aquaculture is human consumption or consumed for the especially for this. They are this commodity development as well as our benefits or as well as for the environmental as well as the economic growth. Okay, so I have come to the point, this aquaculture point or India. So that is we are in India, you are surviving in this country. So that is you are 140 crores of the people is surviving in this country. They have to need for the supplementation of the proteins or the nutrients or balanced diet is required. So here compared to the China is first one of the aquaculture industries. Why? They are consume the aqua industries only. So that's why they are surviving 120 years or 125 years or like this. We are in that our survival year is only for the 65 to 70 our Indians. Why? This is that the food style and as well as for that our lifestyle or the consumption for the unwanted foods. Unwanted food is the food only toxic for us. Okay. So that is we are come to that. You know that the aquaculture, the biotechnologist. What is is any relation to the biotechnology and the aquaculture and marine science. So that is plenty of relation to that. Okay. So in that biosystem, you are depends on biosystem. Any biotechnologist, biochemistry, microbiologist, or marine scientists, they need for one biosystem. Even though for that microorganisms or macroorganisms, eukaryotic system or prokaryotic system, anyone need for that biosystem. So that our ultimate aim is, especially biotechnologists, need for one biosystem. Okay, that is the culture of it. They can grow or eat, then finally you harvesting that final products, right? So that is what is the mechanism behind the development of the byproducts or bioproducts of this. So simple mechanism, your central dogma. So that is mean the three concepts are the worms, metabomes, epibomes, and transcriptomes. For this concept, worms concept given to that your point. Right? So the biosystem need for that, they have the conversion. Simply the biotechnology's ultimate aim is that final product development, your bioproduct development. That's why you need for the biosystem, the conception needs for the transcription process and the translational process. That is the synthesis of that protein. Right? I come to that point as a biochemist, but given to that lot of information. That is, today is given to the lot of information by the Santa process, and yesterday also that prediction that given to the RTR, or any biosystem given to the enrich the matter, enrich the byproducts, enrich the organisms, they are given to a lot of information. So, final concept is the central dogma given to that our byproduct. So, especially in the marine system, that is aquaculture system. The ocean system, they have to be for coastal based, the aquaculture system, mariculture system, the ornamental system, everything. You are come to that idea in Coimbatore. How to culture, how to develop. So simple mechanisms we need for that water. So we need for that some plant, streams or lakes or affair for that. They are need for aquatic system. So you simply call. So the Kilaku came to Sandipingan. Every Kilaku, that is western side, eastern side is ocean. The western side, everything is hillside. How to that the culture is possible? Is possible. Okay, so that's the need for that the culture system. Even you can go for that some microalgae culture. You initiate that Professor Sumati ma'am initiate that microalgae culture system. They are given to lot of information that microalgae need for today is the consumption of that biodiesel, the extraction of the biodiesel or essential fatty acids or essential pigments or essential byproducts or then essential lipids, that is then body unsaturated fatty acids, everything need for us. So today is demand. So the today is demand for this all the things. So the sustainable development for this aquaculture to develop that the all byproducts, especially the fish, shellfish, crustaceans, and the seaweeds, fastest growing microalgae. These all are needing for the soil. Okay. First, I come to that the point of this microalgae culture. So, aquaculture, biotechnology, that is aquaculture in India. Each state is more aquaculture product. So, the most of the states that is so our ocean area. How many kilometers are covered from our coast in Tamil Nadu? Any idea? நீங்களா கடலுக்கு போயிருக்கீங்களா? கடலுக்கு போனது இல்லையா? கடலை என்னமா பாக்குறீங்க நீங்க? ஃப்ரீயா பேசுங்க, ஒன்றும் பிரச்சனை இல்லை. நான் ஒன்றும் பெரிய சயின்டிஸ்ட் கிடையாது. நீங்க ஒன்றும் தெரியாதவங்க இல்லை. என்னமா உங்களுக்கு எல்லாம் நிறைய தெரியும். So you have to given to that what is the information to that behind the ocean, behind the coastal area. என்ன மாதிரி கடலை பாக்குறீங்க? What is the view of the river? Oceans are blue in color is given to the fish. That's all. Your aim. Okay, am I correct? Your point of view, your life sciences students. So only the ocean is blue in color. That is given to that some fish products and fish. No, the ocean is given to that lot of information. all the things we have to isolate, purify from only ocean. In microorganisms, you can culture isolated from the marine sources. After that, you can culture in indoor or field or lake. So next thing, they are culture and on develop the culture system in your own field or own land or in your own laboratory. Like this. Okay, so the research. The ocean is covered our Tamil Nadu, 906 kilometers. And they are coast of that Tamil Nadu, 906. Okay, so 906, but the coastal base area, they have to most of aquaculture industries are available. Okay, which state is more covered in India? The coastal area. So this is Andhra Pradesh. Andhra Pradesh is most of the area that the aquaculture developed from the agriculture. Agriculture and agriculture Andhra. So Andhra is just first the practices in the aquaculture industry. Aqua industry. Why called aqua? So these for that only depends on water. Okay, without water we can't do that aquaculture. Okay. So next is for that. What are the developing the biological system? Now disease resistance, environmental sustainable. This all the things for the previous story given by Professor Santa. What is the global status for aquaculture? So this is China is first, then India is second, then Vietnam and Indonesia. South Asian country is given most important for the aquaculture development. So that is what is the ultimate the product development. The product is exported to the into countries. Okay, so finally the persons are exported to the some other countries, that is export for the our commodities, our products. So that products is export to the MPDA, that is Marine Export Development Authority, MPDA. So that compound matters are export, that nutraceuticals or pharmaceuticals or anything will export to others, especially through that food and other systems. So okay, so this is for that. What are the species you have in India, all the things given to that. So this is next generation byproduct development. So first thing for that, traditional production of the nutraceuticals and pharmaceuticals. Next one is cosmeceuticals, all the things. So this is bioprocess technology. So already you are using that biotechnology is given to the bioconversion. Bioconversion of the simple molecule converted into the major molecule or the macro molecule. Okay, what it is simply amino acids is converted to the protein or enzyme or novel proteins, novel peptides. Like this, they are production of the bioprocess engineering to that aquaculture biomass and the production for this. Okay, this is development of the new economy. So future we are dependent on blue economy. Blue means that is ocean. Ocean dependent on economic development to export to the all over the world. Okay, so this is for the nutrients, then fish aquaculture, then for the final process is for the human nutrition, then domestical animal, then petroleum sustainable products. All the things we for us today. That is, food scarcity is very key. That the food supplementation is very important. Next thing is that energy, bioenergy, that is biodiesel. You are depends on only that biodiesel, that is fossil fuels. Why are fossil fuels are dangerous one? Because the biofuels are created the pollutant. Carbon dioxide is in the atmosphere is more loaded. That is, we are the changes of the climate in our system that leads to that sea level rise or the global warming. Everything created in the marine environment. Okay, all the things is sustainable is very questionable one. That's why we need for this. The microalgae culture, the microalgae is capture the carbon dioxide. The capture of the carbon dioxide is processing for photosynthesis. After photosynthesis processes, the carbon dioxide is converted into the hydrocarbon. That hydrocarbon is a beneficial product for our biodiesel or some esters products. Okay. So next is marine economy. The true economy is given to the fisheries and the fisheries sector is developed for the revolutionary process, development process, especially for need for that collagen and omega-3 fatty acids. omega-3 fatty acids, omega-3 oils, very good for the essential elements for development of the cognizance of your brain. So that is deficiency of the omega-3 fatty acids that leads to the brain insufficient development or inborn errors occurring that the omega-3 fatty acids, then collagen. Why need for the collagen? That the omega-3 fatty acids cognizant development. The protein is collagen is the development of your physical fitness. Okay, that's why we need for the collagen. The collagen and omega-3 fatty acids present in the marine economy. Next for that, conventional aquaculture system that is sustainable, then eutrophication, habitation degradation, and biotechnological sustainable environmental aquaculture. Okay, so this is what goal one for my need. The biotechnology or the aquaculture innovation, the marine microbial biotechnology, you are cultured with marine microbes or microbes, they have to given to the probiotics. Okay, I come to the point. So molecular diagrams and the goal one is microbial biotechnology. What is the microbial? Come to the point. Why are you considering the microbes? Even biochemistry persons also microbes, and biotechnology persons microbes, even microbial genetics person also microbes. Why need for microbes? Yeah. Microbes are bioactive compounds, okay, then easy to grow, okay. Okay, then microbial biotechnology is that microbes having lot of information. So that is biological information. What is the biological information? They have to even do lot of enzymes and the proteins. So that is single cell protein that I was studied in the Coimbatore Science Fair at the time of the 1987 to 90. At the time SCP-1 bus is come to the field of the whiteboard. That is SCP bus, that is single cell protein. I simply refer to that single cell protein. That single cell protein is development for the probiotics and the bioprocessing system. You have the microbes, you introduce the any water system. They are observed all nutrients. What are the nutrients? That is phosphate, then nitrate, and other elements, that is inorganic elements. They have to observe, they are consumed and produces lot of very good amino acids, peptides or enzymes or proteins. Okay, amino acids consume the protein. They are formed the protein. That protein is just a novel product that is called the probiotics. Okay, you develop the microbes, that novel microbes, you put onto that aquaculture system, the consume that particular microbes by the fish or the same fish, they have to disease resistant. Then why that they are disease resistant? The probiotic microorganism induces the immune system of aquaculture organisms. What is that probiotics? Probiotics act as a stimulant for the immunostimulant and vasoactive organs, proteins, peptides, all the things to sensitize the system of the aquaculture fish or aquaculture organisms. They are given to the very good disease resistance. Then only we are using for the probiotics and bioprocess systems also given to the fish shell. That is probiotics having the enzymes. What are the enzymes are present? Like this, protease, cellulase, like this enzyme. They have to digest all the matter through the system of the fish guts. They have to digest all the matter. They are consumed. The feed from that, they are foods for that, they develop the fish system. That is bioblock and the microbial probiotics. Okay. So next okay, what are the marine derived byproducts? So one is for the bioproducts or bioactive compounds good for the antimicrobial activity as well as antioxidant, and then someone for that byproduct enhance the pharmaceutical effect. They are both for development of economic as well as the waste reduction through the microbial consumption. So these all the things for new economic development through the nutraceutical as well as pharmaceuticals. This is for our system. Next thing for that, the high value products for that, the skins for the edible part and the non-edible part in the fish. Okay, two parts are available, edible and non-edible. What is the edible part? Edible part? Fish, right? Okay, then non-edible part. You வெளியில போறீங்க fish waste எல்லாம் அந்த கொண்டு டம்ப் பண்ணுவாங்க. It creates lot of trash. இல்லையா? That product also value products. That waste also given to that value product. That product is for the hydrosan. Fighting. So fighting is today is now one plenty of research are going. That is nanoparticle derived. The hydrosan and fighting derived nanoparticle. Then they are the hydrosan and the collagens given to the cosmetic items. Plenty of they have to the plenty of matters are the meat product, white products, red product, hydrosan. Then another one for the collagen. Collagen is the wound healing protein. What is that? Wound healing means they are injuring of the today's big problem diabetic wound ulcer. Diabetic wound ulcer wound never they are cured by the anything. The wound ulcer they have just with the marine collagen is different to that. They are the tissue engineering need for this marine collagen. They are tissue engineering process. Okay, next the hydrosan and the crustaceans cell walls, cell base. Crustaceans, what's the crustaceans? The salt today's the prawn and the sea crab, they have the cell is loaded lot of hygiene and hydrosan. These waste given to that, they are used in the bioremedial process. They have demineralization, deprotonization, all the things used for the strong antimicrobial activity and as well as used for that. So one concept is the heavy metal remedy. The hydrosan is used to the remedial process. That process is reduce the pollution and the very good economic value for this hydrosan and the crustacean cell base. Okay, next fish protein hydrogenate. This is the bioactive peptide. This is antimicrobial peptides from the fish base and the skin for this. The sustainable development of the efficient for this protein hydrolase, the fish base. So next omega-3 fatty acids. This extraction technology. This omega-3 fatty acid is a polyunsaturated fatty acid. Is a development for the brain health and as well as anti-inflammatory activity in your health aspects. Okay, seaweed based functional foods. So you are to that. So the sulfated polysaccharide. Sulfated polysaccharide you are using the agar. Okay, what is the source of agar? Seaweed. There is agar. So this is sulfated polysaccharide only marine source. Okay, marine sources. So this is sulfated polysaccharide. It's a very good input for this alginate, carrageenate, agar, and the fucoidic. Fucoidic is anti-cancer compounds. The pigments also present in the seaweed. They are induced as the immune system and as well as health aspects for the functional food for this. What is the functional food? You know that functional food. What is the functional food? You know that structural foods are functional food. What is the functional food? Any idea? Functional food is that foods directly go for the liver system is regulated by the particular food or protein. So that food is called functional food. The functional food more of them present in the marine seaweed. So next all the bioactive compounds. The SAR is given with a lot of information for that astaxanthin, phytoxanthin, and the phytoxanthin. This is pigment. So this pigment today is the big problem is that antioxidant one. Okay, you are taking for the fast food. What happens to the system? That is lot of problem is created. Especially the most of the point more hospitals are available. Especially seeing that organ, vessel organ, then the part of 30%, kidney, the liver, brain. Doctors are only சொல்லுவாங்க பயமானது என்ன ரிப்போர்ட் வரும் பயாப்சி என்ன சொல்றாங்களோ இந்த டிஷ்யூ கொடுத்துருக்கோம் ஏதாவது கேன்சர் ரிப்போர்ட் வருமா why That is, they have to our food science given with lot of problem creating or disease inducing or the hypertension, everything inducing process. That's why we got today all is you can take for one gram of oil you consume. There is no any diseases. Why they have to in China we directly take the oil. That's why the company is supposed. And smell up to the market so that is very good antioxidant. The antioxidant then why are always calling the antioxidant? That's antioxidant. You are created the peroxide. That is peroxide. That peroxide is damaged to your cell membrane first. Cell membrane. After that, the cell membrane is damaged, your system is rendered to that all the any pesticides or insecticides or toxic or any matters easily enter into the your cells. Cell can go for that systems are collapsed, the antioxidants, the oxidants are stimulating all disease. Okay, that induces disease. That's why we prevent the accidents by antioxidant. The antioxidants for example, astaxanthin treatment, the single cell example, the glucosamine and phycocyanin. This is all these marine treatments. There are some animals also having that treatments. Okay, that treatments from that protocol they have to given that a lot of information for astaxanthin. Then start getting to the pigments. Out of the pigments are developing that they are consuming copicol. So the copicol they have a lot of isoprenol, that is fatty acids. That fatty acids directly go for that isoprenol units. That isoprenol units are single units given to the your pigmentation of your fish or ornamental design. So that's why they are used for that with the antioxidants development of color. You can take daily 1 gram of algae, 1 gram of glucosamine. You are changing your color. Okay, I dive into that. So next for this. Okay, all the bioactive compounds. The already derived compounds for that glucosamine and various secrets, polyphenol. This is secondary metabolites. These compounds come to the microalgae as well as the macroalgae. So the marine pigments for that, the astaxanthin and microsanthine. So this is support to that anti-cancer effects. So next is that the products come to that. Example from the aquaculture system. So next marine enzymes. So this comes to our point. The microorganisms or macroorganisms produce the normal enzymes. What is the normal enzymes? In group, that person need for the enzymes. Okay, the tannin, you know the tannin, the gene stand. The sun genes area they have to add in color. So that's why the China people they are produces the novel enzymes. They act as a detergent. Detergent means your cosmetics. The need for these enzymes, they are highly valuable enzymes. The improvement of that heats or process, they are using these enzymes. And another one for that. They are using to that some biochemical process also. The enzyme is degraded. The body pollutant pesticides or insecticides removed for that the microorganisms. So next is need for that probiotics already due to a lot of information. This is immunostimulant. The immune system synthesizes for the your micro compounds, especially parasaccharides. This probiotics induces the immune system. What is the immune system? You know that the B cell, and they are produces the immunoglobulins. The immunoglobulin development, and they are go for that immunological reaction, immunotolerant for this. All the things come out from that marine products. Okay, next directly additives for this example. Then discount this example. You can show this. Okay, next aquaculture. This is for the how to the aquaculture development to the aquatic system for the need for the aquaculture. Now nutrients, especially the formulated nutrient operations like this. Okay, this biotechnology starts getting to the next lead. I am come to the prebiotics. So synthetic biotic for this. The immune organs enhance the immune response. So today is the aquaculture system is need for that immunological stimulant or tolerant preference need for this. The beneficial organs, they have to the absorption of the nutrient and the digestion of the nutrients. They are stimulate the immune system. The fighting against the pathogenic microorganisms. So this is the other policies. The nutraceutical development for the aquaculture for the omega-3 fatty acids. The text like this. Okay, this is a seaweed-based like aquaculture production. And the sustainable circular systems for the wastewater, zero waste, that is RAS given to the SAR is given to the lot of information today. The integrated multi-crop aquaculture. So this is entirely different to that. This is integrated form this, the shellfish and fish and seaweeds. This also integrated forming that they are consumed for fish, they are releases the products, there is nitrogen compound, that nitrogen utilized for the seaweed and other plant system. So this integrated INTA is very important system. So I have one more information from that INTA. This is for very useful for that in the field system. INTA, okay, integrated multi-tropic aquaculture system. Okay, next aquaponics is bioplastic production. So the microorganisms, marine microorganisms culture planning, they have to convert to the polyhydroxybutyrate, that is bioplastics. Today we speak about the bioplastics. Plastics is dangerous to us, but the bioplastics is easily degradation process. This is simply go for in that single microorganisms, single cell microorganisms, they are generating a lot of the poly systems are this, then plastics and conditions are developed by the microorganisms. This is simply bioproduct cells. And biofertilizers from aquaculture system, we can culture the marine microalgal culture. They are used for the nutrients. That is, this is go for the biogeochemical cycle. This absorb the nitrogen, then consumed by the plants by the nitrogen through this system. Okay, this is utilization of the model of the biofertilizer aquaculture products. The manual preparation from the bio-waste of the aquaculture products. Next, biopesticides from the marine systems. So this biopesticides for that. The development of biopesticides from the some algae and other matters you are consuming spray out to that. They are disease-resistant plants developed through this system. Okay, next circular economy model in the aquaculture system, that is RAS system, and as well as other the modern technology is use the water system. Okay, zero waste water technologies observe the water and as well as water process. Where anybody is available in the Kerala? You know that Kokali field. You know that Kokali field? No. Kokali field. So this Kokali field means aquaculture as well as aquaculture. So two things for this. The use for the fish is put on waste. The fish is releases the new waste. That waste is absorbed by the plant. That plant is grow well. Then fish survival in the water system of the particular plant, especially dragging. That system is called Kokali feed. Okay, next year, the carbon footprint reduction for this. Today the system is very neat for the carbon dioxide fixation. Reduces the carbon dioxide from the carbon sequestration process. That is bioblock and as well as CETA. Okay, so ornamental products. So the market, you can ultimately all the development after that start getting with the people they are export to the ornamental, especially export the ornamental fish and other ornamental matters through this system. So I might say this. Next, marine refinery concepts. So this is refined for the production of bioethanol, biodiesel, and biohydrogen. This is the organic water. So this is used for the seawater as well as for the single cell, for marine yeast and the other marine single cell, they can produce the amino acids and pigments. So next, scale up for the marine product byproducts. You are the laboratory level to the field level, and come to that marketing level. You use that the scaling up all of this, the quality control and the standardization. After that, you have to for the global marketing giver aquaculture products, especially the biotechnology, marine biotechnology products is go for the export process. The export potential today is developed into the 8 point something. Our GDP is improving that marine quality and the products is exported in our India. So this is some case studies for this. Japan marine biotechnological improvement in this. Next one is Norway, solvent-based bioproduct industries. They are export a lot of omega-3 and other fatty acids and the proteins. The United States for the marine biotechnology is exported this. China is the first for this aquaculture products. The marine products is new for this. So future technologies, what the technologies adapted for the artificial intelligence in the aquaculture system. We will go to the application, the AI in the aquaculture. So I come to that internet of the smart aquaculture system, that is remotely your aquaculture. They have to develop your information is gathered through that remotely, oxygen level, salinity, and pH and temperature. So the nano biotechnology is developed through the marine biotechnology products. So this is for our climate beneficial microorganisms to develop the marine biotechnology approaches. I come to that this is marine biology compounds. This is a big topic. I will not go from that. So this is collaboration. How to collaborate with marine bioproducts development process. So I conclude this. Thank you for your opportunity, Sudhir ma'am and joining knowledge management, and especially for the Santana sir and other faculty members, the Department of Biotechnology faculty members, students and everyone.
Now the session is open for queries.
எதுவுமே தெரியலனாலும் வெளியே கேட்க மாட்டாங்க, ஏதாவது தெரிஞ்சாலும் வெளியே கேட்க மாட்டாங்க. இந்த எந்த இடத்துல இருக்குன்னு எனக்கு தெரியும். Okay. So our ultimate aim is production product development and novel product development through any organisms, even microbes, plants, or the microorganisms also. So that is aim one. நீங்க வந்து இங்க வந்து aquaculture improve பண்ணுங்க, you have to that point to the இங்க யாரும் இல்ல, யாருமே கிடையாது. எல்லாமே எங்க இருந்து வரும்? லேக் சைடு இருந்தா, கிழக்கு சைடு இருந்தா லேக் வரும். அப்போ அது என்ன நீங்க டெவலப் பண்ணணும். நீங்க ஒரு 10 ஏக்கரா இருக்கா, அதுல வந்து 5 ஏக்கரா நம்ம இன்டெகிரேட்டட் மல்டி ப்ரோட்டின் சொல்லுவாங்க. தியாகராஜ ஃபிஷ் அண்ட் aquaculture agriculture process they can water consumption is very less. இந்த ஆர்எஸ் இதை நீங்க வந்து இங்க ப்ராக்டிஸ் பண்ணணும். நீங்க ஹேண்டில் பண்ணாலும் தேங்க்யூ வெரி மச்.
Thank you, sir. That was indeed a thoughtful session. I now request Dr. V. Kalagandhi, Assistant Professor, School of Biotechnology, to honor Dr. V. Manohar. sir. Moving on to the next session, I now invite Ms. Ritika Shankar, Research Scholar, School of Biotechnology, to introduce to the person of Technical Session 6, Dr. R. Rajaram, Professor, Department of Marine Science, Biotechnology University, Tiruchirappalli.
Good afternoon, everyone. It's an immense pleasure and privilege to introduce our esteemed resource person, Dr. R. Rajaram, Professor, Department of Marine Science, Bharat University, Tiruchirappalli. Dr. Rajaram is a distinguished marine scientist with more than 24 years of research experience and 19 years of teaching experience. He completed his PhD in marine biology from Ananda University and currently pursuing BSc in marine science from Aquaculture University, Biotechnology. Over the years, he has built an outstanding academic and research profile in the fields of marine environmental science and biotechnology. So, his core research areas include environmental impact assessment, toxic metal pollution, microbial remediation, marine lifestyle products, DNA barcoding of marine organisms, microplastics and their biomediation strategies, and polar research focusing on metal transit in Arctic footprints. Notably, he has participated in Indian Arctic expeditions in Svalbard, Norway, contributing to global research on environmental contamination and climate metal dynamics. He has published over 139 international research papers in high-impact journals with a cumulative impact factor exceeding 466, index of 36, and nearly 4,000 citations, demonstrating the global impact of this research. He has successfully completed and currently leading several funded research projects supported by prestigious agencies such as UGC, DSC, Ministry of Health Science, PPT, RUSA, and Science. He is a recipient of the DSC Young Scientist Award and the Best Environmental Research Paper Award from the Government of Tamil Nadu. In addition to his research excellence, he has guided numerous PhD scholars, evaluated several doctoral theses, and delivered more than 50 invited lectures at national and international platforms. With his vast academic experience and remarkable contribution to marine and environmental sciences, he continues to inspire students and researchers. We are truly honored to have such an eminent scientist and inspiring academician as our resource person today. Let us join together in extending a warm welcome and heartfelt welcome to Dr. R.
Thank you, Ritika. I kindly request our speaker to commence the technical session 6 on the topic, the impact of microplastic on the polar ecosystem and aquaculture.
Very good afternoon to every one of you. Thank you, organizer, for arranging or following the same schedule as per the schedule we have because after lunch having the class is a little bit difficult. We go end day. So all participants, students will be in the mood of going home. So I have only 40 slides. I will take only 40 minutes. Don't worry. I will leave you at 1:45 p.m. for your lunch. I feel very happy today is the science day. The main motto of the science day is to create awareness to children, students like you. In that aspect, I feel very proud. At least some of my message you can take as for your career to become a research scholar or to do the better PhD dissertation work. I hope I can introduce some of my aspects in the field of your career. So we had yesterday 3 class, 3 deep lecture, today also deep lecture. This is somewhat different compared to previous talk because all the previous talk deals with the aquaculture product related as fisheries and career in all this aspect. This is somewhat different aspect in the area of pollution. How our aquatic organism or aquatic species get polluted, particularly by microplastic. We have certain expertise around 6-7 years and the microplastic aspect in addition to the metal pollution. whatever the knowledge I gained in the area of microplastic, I'm going to share with you with some photograph, with some graphical representation. I am going to share with you, not for any formulas or any theoretical aspects of law. So this is the data of my presentation is impact of microplastic on polar ecosystem and aquaculture. Polar related to that, remaining whatever I covered in other areas, particularly in our coast areas related to microplastic. Only I am going to share. Before that, there are a lot of introduction to microplastic, what is microplastic, how it affects the human health, aquaculture, and all these things I am going to share. Then, whatever my contribution. Down this. So this is about the content of my presentation about this microplastic. Why we need to study the microplastic? What is the overview and the current status of microplastic in our creation, and how it affects the ocean, how it affects the oceanic resources. There are some projects we have completed in the area of microplastic. In the form of case studies, I am also going to share with you some polar expedition photos also with this. Then what kind of solution? This is the era of finding the solution for microplastic pollution. That also how we can tackle the microplastic pollution, how we can control, what is the strategy we can use. All these aspects we are going to cover from this presentation. What is microplastic? Don't think that microplastic means very small, tiny, or microscopic like that. So it is defined very long back. Microplastic means whatever the particle, the plastic particle size is less than 5 millimeter size, those particles are called microplastic. It is defined around 100 years before. That time they defined it is smaller than 5 millimeter size. Our particles are called microplastic. It is non-degradable. It has so many impact on the environment, particularly water, column, sediment, and biota. It can be the larger particle can be degraded by mechanical action like UV radiation, the waves, and it will be converted to smaller. Now, even be converted to nanoparticle. Now, this is the era of nanoplastic research. There are a lot of impact caused by nanoplastic. That also people are studying about nanoplastic and its impact now. So there are two sources of microplastic. One is primary source. Primary source means particle size less than 5 millimeter directly coming to the environment, mostly from cosmetic items. Secondary microplastic means those particles which are larger particle plastic material by waves being the UV radiations slowly get fragmented and become smaller particles. Those particles are secondary microplastic particles. Then this is about the classification of plastic based on the size and its impact. You see here, this is called the megaplastic. The plastic size is more than 1 meter, which affects mostly on marine mammals, marine reptiles. The particle size, the plastic particle size, which is 2.5 centimeter to 1 meter size, the particles are called the macroplastic, and which affects most of the carnivore fishes, omnivore fishes. Those consume the plastic and it affects the gut system and the air system. Then we have mesoplastic, the size is 5 millimeter to 2.5 centimeter, which also affects the marine fisheries, carnivore, and omnivore fishes. Then we have microplastic, the particle size is less than 5 millimeter, and we have nanoplastic also, the size is then from less than 1 micron size particle. Now, nanoplastic impacts are more. There are several research papers that are coming out of nanoplastic and its impact. So when you go to any coast area, any beautiful beach and get the sand and see the sand, you can get this much quantity of plastic. This much quantity of plastic everywhere, not only in beaches or even in coastal areas, everywhere. You can get water or soil or sediment or sea or filter, you can get plenty of plastic particles. So these all are nuisance to the environment. It greatly affects the benthic biota, benthic organism, not only benthic, every individual in the earth, right from microbes to mammals, including human beings, are severely affected by plastic and microplastic. So this is a graphical representation how microplastic and nanoplastics are formed in the environment, how it affects the environment, what kind of toxins released from the microplastic and nanoplastic. This is about graphical representation. about formation, release, source, and the impact of plastic and plastic-based chemicals on the environment and air. So why we need to study as a human what kind of impact the plastic and microplastics are causing to the human health? There are plenty of impact. There are several studies are available about microplastic and nanoplastic impact on the human health. You see here one by one, it causes obesity. Now you see many people, very younger generation people are severely affected by obesity, which is caused by microplastic pollution. Then all kind of factors, short-term and long-term, chronic and acute toxicity, carcinogenic. Now you see here, before 2000 and after 2000, the rate of cancer is increasing like anything. So these all are due to all kind of pollution, even microplastic also one amount of pollution which cause carcinoma, which cause cancer, which cause genotoxicity, which cause developmental toxicity. Then there are a lot of immune suppression, immune activation, abnormal immunity responses, and all microplastic and nanoplastic enter into the gut part and they cause gastrointestinal disorder and affect our gut biota, microbiota. It even disturbs the endocrine gland and endocrine chemicals, damage the lung, and it also causes sterility, reduction in sperm count. Birth rate of infant also reduced nowadays due to microplastic pollution, lot of cardiovascular diseases, breast cancer. These all are the impact reported due to plastic and microplastic. These all are published report, authenticated report. Microplastic and nanoplastic have several human impact. So this is the condition. If you go and visit around our city or in coastal area or any, in every city we have dump. Mostly solid waste. This solid waste, 50% of this solid waste plastic. It leaches so much of chemicals due to interaction of sunlight, radiations. It releases a lot of chemicals. Even the chemical reaches the groundwater. Whatever the groundwater we are taking, consuming, this is also contains some kind of chemicals which are leaked out from the plastic. Groundwater also contaminated by chemical present in the plastic. This is the condition in all cities. So coming to the plastic pollution, the plastic is inseparable with human life. And our day-to-day life activity, every human, if you check each and every human body, plastic in our body, it is inseparable. It is there in the form of button, in the form of pen, in the form of what many material we are using. All material contain plastic. There is no, we are not following regulate the manner to dispose the thing, to segregate the thing, to collect the thing. There is no regulation here. So the first plastic was invented during the year 1862 by Alexander Pottry. So the usage of plastics started in this century only. The over usage of plastic started just 30-40 years back. There are 3,000 plus polymers are available, chemicals are available, which are used to synthesize the plastic. Among the 3,000 plus, 6 chemicals which are more dangerous. These occupy 80% of plastics, like polypropylene, polyethylene, polyvinyl chloride, polyurethane, polytetrapylene, and polystyrene. These are the 6 different chemicals which is used for synthesis of most of the plastic we are using. 80% of the plastic we are using, which is belongs to this 6 different category. So when you go into the ocean, you know that stormwater, tsunami, which was stormwater, tsunami entered into the ocean and bring all the material, waste material present in the land and again take it to the ocean. This is called the tsunami and the stormwater. Due to tsunami and the stormwater, there are 5 major patches of plastic which are floating on the ocean. 2 in the Pacific Ocean, 2 in the Atlantic Ocean, 1 in the Indian and Southern Ocean. They are floating on the sea like a small island. It always interacts with the sun due to photocatalytic reaction, due to wave and wind reactions. This patch, this iron-like sized plastic which are floating on the ocean is get fragmented into small and constantly it releases the dangerous chemical into the ocean. The oceanic organisms are consuming and it gets accumulated in all level of organisms. In such a way, plastic pollution is also affecting the coastal areas, coastal ecosystem, oceanic ecosystem. So this is about current status in terms of waste generation or plastic generation by government of India. So many countries, they are many Asian countries, European countries and Western countries, they are clear with disposing the waste material. They have good system for disposing. They have good system for recycling. Countries like India, particularly populated countries like India and China, you are failed to manage the plastic wastes. As per the recent report says that 80 million metric tons of plastics are thrown into the ocean every year annually. Out of which 336,000 tons are microplastic enter into the ocean every year through the river, which is generated to the land. Even plastics reported anywhere. There are plenty of papers are available. Microplastic and plastics contaminated the entire even we can find it in the Himalayan glacier. Even we can find the plastic and the microplastic in the 11,000 kilometer depth of Mariana Trench and the Sundra. All our ocean floors are dumped with the plastic and microplastic particles. It is everywhere. It is the entire earth is contaminated. Many of the larger organisms, birds and the reptiles, larger mammals, marine mammals are consuming the plastic material. It looks like a feed, it looks like a food items based on the colors, based on the attractive natures, all are consumed and it gets accumulated in the organism's stomach and it is undigestible and slowly the organism dies. So this is there is one recent data says that in 2050, if plastic pollution is continue like this, the weight of plastic is more than the size of total amount of fish or organisms available in the ocean. If you continue for dumping of pollutant and the plastic pollutant in the ocean as it is, in 2050 we can find more plastic than the fishes. This is the situation. This is about the different marine animals, seabirds, reptiles, marine mammals. They consume the plastic material and they death. Particularly, the turtle population is drastically reduced due to consumption of plastic and plastic-based material. This is current status as per our Government of India, Ministry of Environment and Forest and Climate Change report. It says that India alone generated 25,940 tons of plastic per day. This is per day data. Out of it, and then more than 97,000 tons of solid waste per day, according to our ministry report. This is one day. What about recycling? No zero recycling here. Just it is dumping everywhere. So there are another reports say that there are 10 major rivers in the globe which contribute 90% of plastic entered to the ocean. Out of 10 rivers which generate 90% of plastic into the ocean, 3 rivers which are present in India. The major river which is called Ganges, followed by Brahmaputra and Indus. These are the 3 rivers. They contribute more plastic into the ocean. Out of them, 3 are present here, and 2 are in Africa, and some of river in China. So, impact of microplastic on ocean resources. All plankton, even microscopic zooplankton, the they ingest the microplastic and nanoplastic, and it has a lot of impact on them. And since it is a primary food reserve, it involves in all kind of food chain. If plankton population collapse, the entire system will collapse. You know that. It all kind of marine food chain and food web which are contaminated by plastic. Even plastic sink on the ocean floor, and the bottom bowling and the bottom living benthic organisms also get affected by plastic and microplastic. When the plastic or microplastic exposed to UV radiation directly, it releases the toxic chemical which is more toxic than the nanoplastic and microplastic. There are plenty of chemicals they are releasing. All chemicals are carcinogenic. Then this is the effect of microplastic, and particular fish, how the fish, oceanic fish resources get affected with plastic means it gets accumulated with the gut and the gut blockage happen. The digestive problem will come, then immune system get collapsed, then lipid reserve also get reduced, then it affect the entire physiology of the fish, including the photosynthetic efficiency, including the respiratory and the reproductive process also get affected by marine fish which are consumed by the plastic material. So even this is 2017 data, 10-year-old data, plastics are reported in Arctic and also Antarctic. In Antarctic water, in Ross Sea, they reported considerable quantity of plastic in Antarctic water, particularly the Ross Sea. It's well documented. So this is a turtle. 5 different type of turtles are available. These are the migratory animal, reptile, very important in marine food chain. Even you see, leatherback turtle present in the Pacific Ocean has declined by more than 95%. Loggerhead turtle also the population also declined 80% due to plastic consumption and plastic waste dumping into the ocean. These are some of our studies. We have conducted some studies about microplastic in ocean and even land also. That also I can tell you. I am not going to expose myself on research. I am going to tell some of my experience with you. This may be helpful to conduct, to plan your work on microplastic and its impact. For your MSc destination or for your PhD work, you can get some kind of induction, you can get some kind of inducement by seeing all these things. This is the first report. When we discussed with our scholars and our MSc student, we had one student from destination student from Salem. He is from Salem, Western area, forest area. So when we discussed with her, he ready to collect the sample from Salem. So we plan to collect the fecal matter of house growth from Salem in forest area, in urban area, in rural area. More than 100 samples we have collected and to analyze the plastic load in the grown fecal matter. We found that many of the fecal matter contains all kinds of plastic. Here about 7 different types of polymer we have reported because since crow is transferred, whatever the feed available in eat, most of the crow population affected by plastic, they knowingly or unknowingly consume. Many 70% of the samples we found plastic. So it is population. This is the first report published in the occurrence of plastic in house grow in good environment of science and technology. Then this is another paper based with collection of sample, beach sediment sample from entire Tamil Nadu coast, right from Chennai to Kanyakumari. Nearly about 6 sampling locations we have fixed based on different kinds of anthropogenic activity and the seasonal collections we have done in the entire Tamil Nadu coast to collect the sediment sample and analyze the microplastic load based on the different seasons. Unfortunately, we have found all the stations with plenty of plastic load. The seasonal influence also play a major role for distribution of plastic in the beach sediment along the Tamil Nadu coast. If you want to study more on this particular aspect, you can refer this journal. You can have more information about microplastic in beach sediment. This is another interesting study. We have collaborated with one of the leading microplastic scientists in China. We had a good collaboration with 40 different countries. All the 40 different countries, the coastal sediment, we have collected different pieces of microplastic and we pulled all the samples. The China is the lead lab. They analyzed all the particles. Nearly about 14 Asian countries, the plastic pollution trend is similar to India. So we have well reported how the plastic affects the coastal ecosystem in 14 Asian countries, how we play a role in conversion of meso to macro, macro to microplastic. All these findings we have published in this paper in Trends in Analytical Chemistry. Then this is another interesting report we have made in the Kadalur coast. Kadalur is one of the vulnerable coasts, one of the severe polluted coasts in terms of human activity and natural disaster also. During the COVID lockdown period, we sampled in the Kadalur coast about plastic pollution. After COVID lockdown period also, we collected the sample. During COVID lockdown period, we found that the environment is clean, it is free from pollution, free from dumping. After COVID, after 6 months after COVID lockdown, also we made the sample. Since we have a lot of working control force, this also we have conducted. We have found considerable changes in the control force due to lockdown and post-lockdown. On this report we have, these are the material we have collected from control force during lockdown and after lockdown also. We found that human influenced dumping causes a lot of microplastics. Dumping, due to dumping. So this is a review paper. Already I shown this diagram. Nearly about 150 review paper, 150 research articles we have collected. I have collected in particular to thermoplastic pollution. From the 150 research article, we published a paper in Environmental Monitoring about critical review on nanoplastic and its impact on marine ecosystem and marine resources. If you read this paper, if you want, anyone of you want to continue or do microplastic research, if you read this paper, you can get plenty of idea. This particular work, about 20 pages, it brings the information about 100 papers of articles related to microplastic and nanoplastic. This is another interesting paper. Even we have a study in the mangrove forest in Pichawar. Pichawar is the second largest mangrove in India. We have some project in Pichawar mangrove. One student is working for continuous 2 years. We collected several biological samples. Seasonally, we collected soil sample and sediment sample for microplastic analysis. This is the outcome of microplastic in edible fish and shellfish collected from Pichawar mangrove. Based on the whatever available fish and shellfish from Pichawar mangrove was we collected and analyzed the gut and identified the microplastic and characterized the polymers present in the microplastic and reported in this paper. So season here, the finding says that season mainly play a major role for distribution. During the monsoon season, we should bring lot of wastewater from land-based activity and dump it to the Pichawar forest and forest area, where plastic load is high and compared to pre-monsoon, post-monsoon and summer season. So, when you go for policy making or regulation, the filtration of water is important. When the river going to join to the ocean, some kind of mechanical filtration or removing the waste present in the river, which is very important. Otherwise, dumping will grow like anything. These are all the data about plastic characterization, microplastic characterization in water and the sediment sample collected from Pichawar mangrove. This also we have communicated to physics and chemistry at the journal or the daily journal in pollution. These are some of the data. So the working ecosystem, very important ecosystem. It plays a major role for biodiversity maintenance, land protection, which is called as mangrove ecosystem. Such a wonderful mangrove ecosystem also severely affected by plastic and microplastic pollution. So this is about 25 different species of finfish and shellfish we have collected from Kadalur coast. This is highly polluted coast based on the different feeding habits like the carnivore, herbivore, omnivore, finfish, shellfish, crabs, mollusks, whatever available fish in the Kadalur coast, we collected and analyzed the microplastic load based on the season. We found a considerable amount of plastic load and the polymer in all the fishes. So very recently, 2 years back, even microplastic not only affects water column, sediment column, surface sediment, even it entered into 1 meter depth. We have fixed around 5 different stations based on different kind of anthropogenic activity like industrial activity, harbor activity, fishing activity, pilgrim activity in the entire Tamil Nadu coast like Chennai, Nagapattinam, Bombay, Kanyakumari. All the area, seasonally we collected the sample in sediment at the 1 meter depth, 1 feet depth, and analyzed the load in top column and 1 feet down depth, microplastic load. Even microplastic and plastic material enter into the 1 feet depth also. This wonderful finding we have published recently in Marine Pollution Bulletin. Even at the depth, the plastic material present in 1 feet depth get fragmented by microbes present in the soil column. By mechanical action also get fragmented. which all are proved by STEM and STEM images and all. How the plastic which are present in the 1 feet depth, how it get fragmented, how it changes, all are documented in this paper. So microplastic is everywhere it is present. If you go with the occurrence, distribution, and the impact of microplastic on any resource into any agency for funding, they will throw our proposal. Everywhere is there. What is the solution? How you can control? This is the current area of research. Everywhere is there. It is polluted. Already there. This is over. Identification, characterization, distribution. What is the solution? You bring the solution. You will provide one for anything. There are many methods out there for preventing controlling the plastic pollution. Chemical method, physical method are there. Or the chemical and the physical methods are dangerous methods. They will convert one form of toxic into another form of toxic. There is no solution. All physical and chemical methods. Best method is biological method. We can use any kind of biological method which will convert, reduce the toxic produced by the microplastic, or convert the toxic form into low toxic form, not 100%. So in this aspect, we are doing some kind of research, particularly microbes. They play a significant role for converting the plastic material into non-plastic material. We have surveyed several plastic dump in the coastal system, even in urban system, and identified several bacterial strains. One of the bacterial strains we identified is Bacillus cereus. Within 40 days of experiment, we should reduce the toxic effect of plastic up to 14-15%, not more than that, naturally. But we need 100%, at least 80% degradation efficiency. But naturally, within 40 days, this particular strain converts or detoxify 40%. This is the first report. Previously, from Bacillus cereus, 12%, 11% degradation efficiency reported. This is the first report, 40% degradation efficiency reported. If you go for some kind of genetic modification, some kind of molecular to improve the efficiency, we can achieve. This is one of the area through the microbes, through the bacteria, we can control, we can remediate the soil or water. This is a wonderful report we have about the size of the total environment. If you want more knowledge on this, you can download this paper and read it. Then another interesting finding, next step. We have around 14-15 strains which have good efficiency to degrade the plastic and the polymer. From the 14-15 strains, we selected 3 strains and developed a microbial consortia. All the 3 microbes in the consortia, and we introduced all kind of 4-5 different kinds of polymers, and we introduced the microbial consortia and studied the efficiency of degradation. We found that good efficiency. The microbial consortium has good efficiency to degrade multi-polymer, not one polymer, multi-polymer. We have reported, and this particular work also we submitted to the journal. Revision also submitted. Very soon it will get accepted. So through the microbial bacterial consortia, we can degrade the polymer microplastic in the environment. So these are the initial steps. Current situation, we need a technology to spray something on the dump. Within 10 days or 15 days, entire dump become nil. Such a technology we need to control the pollution, microplastic pollution or solid waste pollution. That means next year people everywhere they are thinking about microbial product, enzymes. There are enzymes secreted, the enzymes through the enzyme only we degrade. We need the enzyme. Some enzymes there are amylase based enzymes are reported. We need to extract the enzymes from the microbes and spray on the dump yard, and within 10 days or 15 days the dump yard becomes digested by all the enzymes and become nil. Such a technology we need. People all over the world, they are trying to identify the potential strain, microbial strain, and isolate the enzymes from the microbes. And it is in the lab in stage level. Next stage, they produce the enzyme in mass level and go into apply in the field. If you have such a technology, we can clean our lab. So, for the past 4 expeditions, 2 air participators, one is my student, another one we got a sample. We have good experience in Arctic also. Antarctic, there are several types of microplastic are reported. Arctic also we have reported. These are some of the photographs. The Arctic fields, we have a Svalbard, which is international research station. Nearly about 40+ countries doing research in the Arctic region, particularly the Svalbard, which is very close to the North Pole. India is also one of the countries. They are doing Arctic research since 2008 onwards. We are monitoring the Arctic fjord by heavy metal and microplastic pollution. Fjord means nothing but doing what is fjord, then only you can understand. Here we know that river mouth or estuary. Estuary means river join the area in the ocean. River and the sea meeting area or river mouth is called estuary. It is very common in all the tropical region. Like India, we have many estuaries, many people. If you go to the polar region, particularly the Arctic and Antarctic region, we have a fjord ecosystem. Fjord means nothing but this is called a fjord. All the three areas covered by mountain. One area is open to the ocean. This is called like the estuary only. Fjord. This is called fjord. This fjord is called a storm water. This is called glass water. Many people you can see from this fjord. This is very sensitive ecosystem. So you see that coral reef, mangroves. These are all sensitive ecosystems. They have a lot of benefit from the ocean. Like that, in polar region, fjords are very important ecosystem. The melted water comes to the fjord, and the sea water comes into the fjord, and it is a mixed fjord. It has different biodiversity, different plankton, different microbes. And here only Arctic research, Arctic marine research are going on. For the last 4, so from 2020 onwards, we are monitoring this both the fjord. We have a lot of data on heavy metal and microplastic. These are all the sampling locations. We have collected all the water samples, sediment samples, plankton samples, from the many biological samples also we have collected and done for microplastic and also heavy metal studies. There are 3-4 papers we have already taken. Another 3-4 papers we have data. Once model is working, there are very interesting applications going to come in this area very soon. So we monitor in the both the fjord. We fixed a 10 station, 20 stations. We collected the surface sediment, surface water, bottom sediment, and bottom water also we have collected and analyzed the plastic load. Even bottom water, surface water, soil, all are contaminated with the polymer. These are the distribution of microplastic in Arctic water, not only the surface. Arctic, this region is very region. No human activity. Only 40 countries are doing research. 40 research stations are there. How plastics are entered into this area? There are water current, oceanic current with the transport. Atlantic water intrusions are there. They are bringing all the waste present in the Atlantic water into Arctic region. Surface transport through the area, through the wind, they are transported, they are deposited. So even Arctic and Antarctic region, whatever the atrocity we are doing here, it reflects the polar region. Polar region is very sensitive region. All kind of DDT chemicals, all kind of pesticides, all kind of metals reported in polar region. So in that way, microplastic also contaminated in Arctic water, not only the surface water, in bottom water. So there are 2 years we continuously monitor Arctic water contains microplastic. Then last year, water only present microplastic, sediment only present microplastic. Then we think that what about the glacier iceberg? Iceberg also we have analyzed. Iceberg it was found 100 years, 200 years before. From the glacier we break down and document on the ocean. We collected iceberg and analyzed very recently. Iceberg also contains microplastic. That means that the deposition, transportation happened very long back. These are the plastic load present in the bottom of Arctic fjord. This is a vertical and horizontal distribution of microplastic. Even surface water, and bottom water, and bottom soil get contaminated by microplastic. We have analyzed all, and it is also on the communication in physics and chemistry of the current is review. This is another interesting report I told you. Heavy metal and microplastic liberation from sea terminating glaciers. That means in iceberg, we have collected iceberg samples which are close to the glacier. This is not a glacier. There are 6 glaciers we have identified from both of you. Glacier means nothing but a river, like it is like a river meeting point in the fjord. It is fully frozen, due to the tides and wave action, this area get the ice get break and ice break come to the fjord. Those icebergs we have collected for heavy metal and microplastic analysis. Heavy metal is also present, microplastic is also present. You see, these are the methodology. One of my students participated. We collected. This is called iceberg. This is the glacier meeting point. So this area we have collected only iceberg through the net and allow the iceberg to melt, and the melted water 5 liters we have taken for all kind of studies, heavy metal and microplastic studies. This is the entire graphical representation of methodology. So this is called the iceberg, as belongs to microbiology, biotechnology, and zoology student. Iceberg means we can see only 10%, 90% it is invisible. A larger particle. From that we can collect 10 kg or 50 kg of iceberg and melt the water, allow the water to melt. From the melted water we analyze. These all are the plastic material which are present in the iceberg. That means these icebergs are formed several years before. So due to fish netting activity, fishing activity, and the transportation and aquatic water intrusion through the aerial route, it enter and they deposited in the Arctic. So these are some of our experience about microplastic. How this microplastic is affected by aquaculture system and aquaculture species. Even if you are having plastic, I said, plastic is inseparable. All the fishing nets are plastic made. All the cages are plastic made. We have fiber board. We have many mechanized board which are made up of plastic and fiber. All plants are fiber. It is inseparable. It is available. So through this activity, through dumping of waste and solid waste and the plastic of the ocean, knowingly or unknowingly, we enter into the ocean and oceanic animals, particularly aquaculture species or aquaculture fish species, consume it and it affects all kind of health impact, health problem. So there will be accumulation of plastic in the gut and it will reduce the fishing, reduce the feeding efficiency, then slow the growth rate, sometimes inflammation caused by microplastic accumulation in the fishes. Then muscles like the bivalves, oysters are vulnerable to microplastic pollution. This is a filter feeder. Not only microplastic, all nanoplastics are entered into the bivalve waste reports and they get severely affected since it is a water-holding animal. Then many toxic, when it gets accumulated in the gut, it releases all kind of toxic chemicals like even microplastic and heavy metal, PCB, the PAH, these are the emerging contaminants which also get accumulated due to accumulation of microplastic in the gut. And if it damages the gut, there are injuries, there is the chance of injuries from the gut also. It alters the microbiota in the gut, it will increase the vulnerability to cause many other physiological and immune disorders of fish. Then it also affects physiological and cellular effects like oxidative stress, inflammation, liver damage, and hormonal disruption. Altered metabolism also causing the fish which is consumed by plastic and microplastic. A reproductive impact like reduce the egg production rate, lower the hatchery rate, abnormal larval development, and hormonal disruptions. These are all caused by microplastic dumping or accumulation in the fish. Then overall, it reduces the growth and reduce the survivability of the organisms which are interested to culture. These are all the impact of microplastic consumption of fish or aquatic species. Then these are some of the remedial methods, remedial sources. The first one is we can control the plastic and microplastic from source itself. European countries are doing that. Even in all the reserve forests, we are doing that. We cannot do anything in the forest if we go to the forest now. Land with here only is a problem. So we have to control the plastic and microplastic in the sourcing side. That is better thing. Second thing, we need to have policy, strict policy. If you don't dump anything in the reserve area, you will be put by fine. Your vehicle will be seized. cancel your license. Such a strict regulations and rules are needed. Overall, awareness is very important. Awareness is everyone. Hereafter, you try to reduce the usage of plastic material. Whatever you are buying the material, you can use maximum time and done in a proper way. Reduce the item of using the plastic material. Avoid the single-use material. Carry some bag. Reusable bag. If you go to Singapore now, particularly Indian, in Tamil, they used to call Manjappai. You may be here at Manjappai. Our ancestors, they followed everything. Even up to 10th class, I used to take my books in Manjappai. How many pens you are taking? You just think about it. See, in my young age, school age, I used to have one family pack for 5-6 classes. Now you see, every day, everyone is carrying one bag. How you are going to dump all these things? Where it goes? It causes pollution. So you buy one pen, at least you use like use 1 year or 6 months. Such a habit you can develop and reduce the waste item. Otherwise, you see the medical history before 2000 and after 2000. After 2000, diabetic cases are more, cancer cases are more, blood pressure cases are more. These all are due to, not only due to pollution, population explosions are there, food are there. Pollution also a main role. So such a thickly populated country, as a human, as a citizen of India, as every one of you, our mind, we have to change our mind. We have to minimize our the material commodity that we are using daily. Then only we can bring a good solution. So removal method, degradation method I have already told you. Then resource recovery. So already polluted. How we can recover those resources like fishes, mammals, even lot of terrestrial mammals also get affected by. Even you see there are photos I have in couple of posts when we go for sampling. Yeah, goat is coming and eating the polythene that only the mutton we are enjoying every week. The goat is eating the small size fishing net. All terrestrial animals, marine animals get contaminated by. So resource recovery is very important. Apart from that, as this is I feel that this is awareness sector. I want to tell you something about plastic and the plastic impact. So at least a few of you can change your mind to use the material less and reuse the material. You avoid certain material to use. This kind of changes we have to bring up in your mind. So for the past 3 years, with the help of National Center for Coastal Research funding, we are conducting coastal awareness program, international coastal awareness program in Nagapattinam. All our department students, scholars used to go to Nagapattinam and clean the beach and create some awareness to the people by showing some rock, some slowly shouting. We are doing some kind of awareness to create about plastic impact and microplastic impact on the ocean and the people. These are some of our activities for the past 3 years. It is a continuous. Every year we used to do. Our people, our students are doing. These are some of activities we have conducted. So with this, I thank each and every one of you. I also would like to thank the organizer and the authorities of the Security College, my colleague Dr. Santam, for giving me the opportunity to discuss with some of my class, some of my work in microplastic. If you have any questions you can have, or if you want to do any kind of microplastic related work, you are always welcome to our department. We are ready to help you, and support you with this. Once again, thank you all. Also, special thank to myself who is sitting here. It's the first time I am bringing here, bringing to show my activity. In school time and all, I never taken him to my department and all. It's the first time. I also thank to Nitish for sitting here and reserving our department and my activity. Thank you.
People not to use plastic bags and one-use bags. Why not the government is not taking initiative to stop the factories producing plastics?
I also don't know the answer. Every time I used to think.
Yes. So
There are-
being a scientist who is working in microplastic, you might represent to the government. Right?
Yeah, I have submitted two reports. On the two reported, given my recommendation to ban the industry, the entry the biodegradable. Even Coimbatore, I hear that there are some industries started biodegradable plastic using glass top.
Industries, they are using the biodegradable plastic. Within 3 days, it will degrade.
Yes, here it is available, but we need to encourage the cost to play a major role. Even if you pay 10 rupees, they give hundreds of carry bag. For one biodegradable bag, they have to pay 10 rupees. The cost is playing a major role. Even the government has to give some kind of importance to reduce the industry. Even our in opposite of our industry, we have ATR plastic factory. We are manufacturing anything. So government also will take some steps. I got one project from the Tamil Nadu government. I given their companies. At least 50% they can reduce.
Thank you very much for the awareness talk about the microplastics. Sir, regarding the hot drinks taken in the plastic cups or paper cups coated with polythene, what kind of danger we have in the daily use, and how do we analyze that what kind of dangers means chemicals or microplastics? As a research point of view, what kind of analysis or methodology to be followed in this regard, sir?
All the paper cups, the polythene coated cups, because they need to resist to water. The chemical is not very extended. It is also a potent toxic. If you want to do any kind of research, you can use warm water on the plastic cup and take the sample and go for physical analysis. Show you how much quantity of damage and other plastic based chemicals are. It is unconvinced. This chemical. It is not purely plastic. This paper. The paper is coated by phthalate.
So we have to analyze the water.
Water. Water with hot water.
Hot
water on the cup. All the chemicals in the cup it leaches out. That water you can analyze.
So we have to analyze which is yes.
It is there. It is known. It is medical therapy. It is known to personal care. It causes cancer.
The water, the cup things. So the one more thing, the liquid batter is used in the polythene covers. So as the bacteria will degrade the plastics, the liquid batter contains a lot of bacteria. So whether that will degrade and it will cause microplastic pollution. So liquid batters are available in the plastic covers nowadays.
That is strictly avoided.
Because others are not be sterile, but liquid batter they will contain microbes. So microbes will degrade plastic. So what will be the concept?
Microbes degrade the plastic and also it will produce lot of chemicals. When degradation happens, larger molecule becomes smaller molecule. There are smaller molecules which is more toxic than the larger molecule. It is strictly avoided. We have to use the plastic. But if you go to Chennai, the disposable one how they are using this idea that they are boiling. They are boiling. When boiling, when it reaches the temperature, it heats out the temperature. You can do the heat only. If you eat the heat, you are eating poison. It's strictly avoided.
I have a question.
Good afternoon, sir. It was a wonderful session with a lot of evidence. Sir, I want to ask this question. There's a lot of content on the internet with respect to plastic eating bacteria. plastic degrading bacteria. Is it a myth or a reality?
Yeah, it's so we have a bacillus series, certain microbial consortium. We can degrade, we produce, it is not eating microbes, it produces certain chemical, certain enzymes, and enzyme degrade.
They will degrade and
when you see the bacteria, not eating bacteria,
yes.
degrading bacteria by secreting certain substances.
Certain enzymes will disappear.
Microbes, particularly the plankton, ovipods, they are agents. They are eating small particles.
So I said the initial sessions.
So bacteria are they produce certain chemicals.
Okay.
That if not chemical enzymes, that the enzymes degrade.
Those enzymes degrade. So the forms that are obtained after degradation, do they cause pollution or?
If you use microbial system to degrade, it is not much harmful. But of course, we have harmful, little harmful. Even small product also really toxic. We use physical method or chemical method to reduce or degrade. It is highly harmful. Even people, small countries, they are collecting all the plastic and the melting with the melted plastic. They are making road.
Yes.
They really get this. So those kind of physical method or mechanical method is required.
Because for the existing plastic, can this be implemented on a commercial scale?
So that is very big question. How we can such a huge quantity is there. We have to collect and what we have to do. One side production is going on, one side continue going on. There is no recycling. Either we have to do something, and then we have to no way to do. At least we have to stop the production. That also not. We are going very seriously. Say, the production is going like anything. Nothing also going like anything. We have to do anything. Anyone, either we have to stop the production, or we have to collect all these things and do some research. That is a big issue. The student, the young mind, have to find some solutions.
Thank you for the thought-provoking session, sir. I now invite Dr. A. Ramatilaka, Assistant Professor, School of Biotechnology, to honor our resource person of Technical Session 6, Dr. R. Rajaram. Dear participants, after your lunch, we have arranged for a validity session. Since no one has got enough time for discussion, before the validation, we will be giving 10 minutes time for asking questions to the panel. If you have any queries, and if you haven't asked during the time of their presentation, due to the lack of time, if you have any queries related to this aquaculture or this 2 days event, you can very well ask during that time. Thank you.
Thank you, ma'am. We shall now break for lunch and reconvene for the biology session at 2:30 p.m.
Participants, please know the feedback forms are given to you. You can fill the feedback forms and submit it to the MCS after your discussion.
ரொம்ப இன்ட்ரெஸ்டிங்கா இருந்துச்சு.